Okay, you can go ahead.
Good afternoon to one and all present here. I, Kate Patel, General Member of IIC, welcome you all for today's event. Parishodhana, an industry-defined problem. Before we gear up for the event, I would like to introduce the motto of IIC. Institute Innovation Council, that is IIC, is an initiative started by the Government of India which organizes events and seminars that are related to startups, innovation, entrepreneurship, and intellectual property rights. The main aim of the committee is to systematically foster the culture of innovation and build a startup ecosystem. Parishodhana is an initiative taken by IIC BNCP to nurture young minds who have the potential to transform conventional concepts necessary for the upliftment of the pharma business. It encourages students to apply fundamentals and principles of pharmacy to solve technical problems faced by the industry in a competitive manner. Before starting the event, I would like our principal, Dr. Munira Momin, to enlighten us with a few words. Over to you, ma'am.
Thank you, Heta, and good afternoon, everyone. It's wonderful to be with all of you, and Parishodhana especially is very close to my heart. I always believe that pharmacy students, being a professional, I do not want them just to be a bookworm or knowing the theoretical concept. What we are looking at, and we always work at, is understanding those basic concepts and implementing them into practicality. And we realized that Parishodhana, since its inception in 2015, when we started thinking of starting the entrepreneurship cell, and so Aarti Madam, Sandeep, all of they have taken entrepreneurship and innovation activity at institute at a different level. And when we see our own alumni coming to judge and help us as a family member. Vinny, thank you very much. And you yourself being an entrepreneur, I'm sure it's going to be a huge motivation to our student and inspiration to our students. Your reasons, your ideas, sharing your concepts and your thoughts will definitely make a huge impact. I also welcome our second judge, Dr. Shripad. Thank you very much for your, and we really value your time as well as efforts and inputs which you will be giving to our students while judging. And I'm sure your experience is going to make a huge difference and is going to give a motivation. Uh, Prabha Madam, you have been always there, and she being a pro student faculty, Vinit would be there to endorse this. That Prabha Madam is always a pro student faculty. Thank you, Prabha, for being a judge for today's event. And students, this is an opportunity where you will understand the practicality of your theoretical knowledge. I'm sure those who have taken part, they would understand the concepts which you have studied, the industry defined problem which we have given to you, what you have understood in that, and the way you come up with a solution, and you come up with a sustainable solution. So whenever I talk to student, I always understand that pharmacy student, when they have to put up their proposal, their ideas, it has to have a business potential. It has to have sustainability, cost effectiveness, and at the same time, it should have market value. May it be pure industry defined problem or it is a patient oriented issue. So I'm sure today's IDP participation into IDP is going to open up a new avenues for a lot of our students. So thank you once again, all our judges, for accepting our invitation and giving your valuable time. And thank you, Sandeep, Aarti, and our backbone is our students. All the student volunteers, thank you very much for organizing and coordinating this entire event. And I always believe since lockdown that this online has become a new normal. I, at this point of time, do not feel at all that we all are sitting at different places and we are coming together on one platform. I feel the same way, like how 2015 to 2021, 20, all Parishodhana which we have conducted in Juhu Jagrati Hall, I still feel we are in Juhu Jagrati Hall. So enjoy this new potential, new normal, of being online. And when I say enjoy, what I want all the participants to do is be attentive, listen to your counterpart, listen to all the teams, learn. Do not just listen or just for the sake of listening. Listen for learning. Listen to all the experts and learn. I'm sure all of you have a potential to be a good entrepreneur. And innovation is something which is the hardcore element, which is there when you want to be the entrepreneur. So thank you once again, and I'm sure all of you are going to have a wonderful time. Thank you, Sandeep, and thank you, Heta. Over to you, Heta.
Thank you, ma'am, for your precious words of wisdom. I would now like to ask Dr. Sandeep Zine to say a few words.
I just lit on. Okay. Yeah, very good afternoon to all of you, respected principal madam, judges of an event, Mr. Sripad and Prabha madam, president of IIC BNCP, Dr. Arti Prabhu, and Mr. Vinit Shaha, then core driving force of our own IIC students and dear participants. So today we gather over here on the occasion of 6th Industry Defined Problem Contest, Parishodhana 2022. And I would like to draw your attention that this time and the first time we had opened up this contest for pharmacy colleges of Mumbai region. And we are glad to inform you that a total 27 teams participated in this unique event, industry defined problem contest, and out of 27, 7 teams were from other colleges of Mumbai region. So we, IIC BNCP, really appreciate your active participation in this unique event. As told by Heta, that Parishodhana is an initiative of the institution's innovation council of BNCP, where students get encouragement to apply the fundamentals and principles of pharmaceutical sciences to solve the technical problems maybe faced by the industry in a very competitive manner. So BNCP always strives to make
Very good. No,
a
I'm done. No,
conscious
he
effort
makes some comment.
to acquaint students with practices followed in the pharmaceutical industry
But
setup,
it comes when I
which
speak
are believed
continuously,
is critical to
people will
their
come
training
that I have
and
something in my
shape
mouth.
them towards becoming successful pharma professionals.
But and then you
I think Madam mic is on. I just put it on off. Okay, so we appreciate the time the judges took out from their busy schedule, as well as our sponsor, Mr. Vineet, to join us for sharing your insights, wisdom, or maybe the expertise. So we will always remember your interaction with students. We challenge them as well as guided them in their journey to become a successful professionals. So IIC BNCP also appreciates the sponsorship of VRW Rubber Industries to promote the young minds on the road to the entrepreneurship. So IIC is always grateful to our principal madam as well for continuous support in every initiative of IIC. So without taking too much time, I think we can go ahead on the next segment. Over to you, Heta.
Thank you, sir. I would
Sandeep,
also.
can I comment?
Yeah,
Can
yeah,
I
yeah,
comment?
madam. Yes.
All right. Vinit, I missed mentioning your sponsorship. So thank you, Vinit, for sponsoring this event. I'm sure it is going to have a good impact on our students. And I also welcome all students, those who have participated from other pharmacy college. And it's a huge response. I'm sure it's going to be a learning event for all of you. Yes, over to you, Heta.
I would also like Aarti ma'am to say a few words now.
I think Aarti madam is not there, Heta. You can continue.
Okay, sir. Thank you.
Okay, madam I join. Madam I join. Yeah.
Thank you very much, Heta. Uh, this is another round of Parishodhana. And welcome, Vinit. I'm meeting you for the first time here. Uh, Thank you for your leadership in sponsoring this event of your own college. Let me say that at the outset. All the students, thank you for your overwhelming response. We do this for you people because we want to make your industry ready. We want you all to understand what are the real problems faced by the industry. But I mean, ताली एक हाथ से नहीं बजती है. So from your side, you all are actively participating, contributing, and that's what makes this event a success. So best of luck to everybody. There are few teams which have been shortlisted, but all of you all have made a superb effort in applying your brain in preparing the presentation. So kudos to all of you all, and good luck to the shortlisted participants for the event. Over to you, Hita.
Thank you so much, ma'am. I would now like to welcome our guest. So sorry. So the sponsor for today's event is Mr. Vineet Shah, who is the MD of VRW Rubber Industries Private Ltd, manufacturer and exporter of roller covering stripes, industrial belts, silicon products for pharma sector, and generally rubber molded and extruded products. Over to you, sir.
It's a great concept. As ma'am said earlier, it's one of the most important skills for an entrepreneur. We look forward to Japanese companies usually for principles like Kaizen, Pokayoke, and things like those, which focuses on incremental growth on daily basis. It's cost-effective, innovative solutions that an entrepreneur is supposed to come up with regularly. That's what an entrepreneurship journey is about. So I hope this competition helps the students to identify those skill sets amongst themselves as well as grow on them and probably turn out to be great industrialists, manufacturers, entrepreneurs in the future. So all the best to all of you all. And it is a pleasure from the side of VRW to sponsor this event to help increase the entrepreneurship drive in our region, in our country. So all the best to all of you.
Thank you so much, sir. I would now like to welcome our guest and judge for today, Mr. Shripad Marathe. He's a pharmaceutical research and development professional with about 16 years of professional career in various esteemed organizations like Johnson & Johnson, GlaxoSmithKline Consumer Health, and Rubicon Research Private Ltd. His expertise lies in the sector of formulation and development, including NC development and consumer health products. He is currently working at Cal Scientific India Private Ltd in a technical operation role, providing technical support to Janssen's external manufacturing sites. He is passionate about design of experiment and statistical analysis. He has won several key organizational awards throughout his career, including Vice President Research Award at Johnson & Johnson and Innovation, Performance and Trust Award at GSK Consumer Health for his excellent technical contributions to the organization. His hobbies include teaching, reading, and traveling. So, moving on to our judges for the day, we have Dr. Prabha Singh. Completed postgraduate diploma in patent law and practice in intellectual property from SVKM's NMIMS Institute of Intellectual Property Studies, Vile-Parle. A patent has been granted on topic of floating micropus osmotic tablet by Dr. Prabha Singh. She completed her PhD in pharmaceutics, awarded degree for completion of research project entitled Formulation Development and Assessment of Controlled Release Gastroretentive Systems of Famotidine in combination with pantoprazole for effective gastric ulcer treatment from C.U. Shah College of Pharmacy, SNDT Women's University, Mumbai.
Heta, you can just introduce me as your teacher and faculty. It's all right. Okay. I do not need to go for lots.
Okay, then ma'am is my pharmaceutics teacher. I would like to introduce you that way.
Yes. So
Okay,
we welcome our guest judge Vineet. Thank you so much. Okay. As a faculty of BNCP, I would like to personally thank you. Okay, it is always nice, our alumni. It's always, a wonderful thing, and it's a pleasure from inside. When we see our students doing so well and giving something back to the alma mater, that's the best thing. And you are going to inspire so many to do it for us. So it's a really thank you so much, Vineet. Thank you, sir, for agreeing to judge the event. And we can start because everyone has, all students are ready. Then let's start, Heta. Okay?
Yes, ma'am.
Yeah. All right.
So before starting the event, I would like to instruct a few rules. First, participants are requested to join the meeting link with their assigned codes. If anyone joins through their name, the entry of the team won't be permitted. Second, every team will be allotted 10 minutes to present their solutions, which shall be followed by 2 minutes of Q&A session. Now I request only the team who's been presenting to stay in the meeting and rest to leave.
Okay, first,
So
initially, I think we judges will talk, isn't it? And then we will take entries. What do you suggest?
Shri,
Go ahead, Sandeep.
Shripad sir, please, if you'd like to address us and our students.
Okay. Yeah.
Thank you, sir.
Okay. Uh, First of all, thanks a lot for giving this opportunity to interact with students and to see their skills developing into this area of problem solving. That's I was really impressed to see that we are trying to make our students ready for the industry, because this is something which is important when they actually go for their actual work in the field. It's always different from what we teach or what we learn in academics versus when it comes to the actual experience. So really excited to see all those participation and see their ideas in the presentation. So good. Let's hope that we will have a very fruitful session today. But we would have some suggestions at the end probably as well to the students, because once they finish off with their presentation, we would understand more what was their thought process, how they went about the problem, and how they presented. So let's have a small, maybe say feedback session at the end as well, to give that feedback to all the participants. So thanks a lot for inviting me, and look forward for a very interesting session.
So Heta, I would like request you to just put the rules in front once for to all the people, if they know, students know, or just once revising the rules of their presentations once again, and then let's
okay,
start.
ma'am.
Yeah?
Okay, ma'am. I'll repeat the rules again. So before starting the event, I would like to instruct a few rules. And the first rule is participants are requested to join the meeting link with their assigned codes. If anyone joins through their name, the entry of the team won't be permitted. Second, every team will be allotted 10 minutes to present their solutions, which shall be followed by 2 minutes of a Q&A session. Ma'am, can I start with the first code? Okay. So let's start with the first code of the event, that is IDP 12.
Is my voice audible to everyone?
Yes.
Yes, we can hear you.
Yes.
Good afternoon, everyone. My team code is IDP12, and the problem statement given to me is that is an antifungal cream formulation which contains sodium sulfite as an antioxidant, and the assay of the antioxidant is a critical quality attribute, and this product is to be commercialized into two pack sizes, that is 5 gram and 10 gram, in aluminium collapsible tubes. On stability, it was observed that there is a significant drop in antioxidant concentration in 5 gram pack as compared to 10 gram pack at each stability time point. So uh, we have to find out the what is the cause for this problem and how to resolve this problem. This next slide. Next slide. My slides are not moving. Okay, so the cause of the problem, moving towards the cause of the problem. So as the antioxidant given, uh, I want second slide to be shown. Second slide, please. So the cause of the problem, moving towards the cause of the problem. So the antioxidant here is a sodium sulfite, which acts as a reducing agent to show its antioxidant activity. So as it has a low redox potential than the adjuvants which it is going to protect. So the mechanism of action is like this for the sodium sulfite. So here the cause for the problem is that can be there can be leaching of the aluminum metal ions from the collapsible tube, and it can react with the sodium sulfite antioxidant to form sodium sulfite uh, aluminium sulfite. Please next slide. Can you please put next slide? So as shown here, there can be a leaching of aluminum ions, and as it has a higher oxidation state than sodium ions, it can exchange it and form aluminum sulfide. There can be this possibility because it has higher reactivity with metal ions, sodium sulfide. And there can be drop in the concentration. Please, can you put on second slide? Second. So the aluminum can act as the catalyst for this reaction. So due to this, degradation can occur of sodium sulfide. And here another problem is that there is no comparable means as there are two sizes of size pack, that is 5 gram and 10 gram, but the significant drop of antioxidant concentrations is seen significantly in 5 gram pack. So the reason for this can be because there must be the concentration of antioxidant can be the same in both the size tubes, but as the amount of the antioxidant is lower in 5 gram tube, so it takes less time to react. So we can observe the drop in the concentration easily in 5 gram pack. So the solution to this problem will be discussed by my teammate next to her.
So as we discussed the causes earlier, now we are going to discuss the first solution to the problem, that is lacquering. So what is lacquering? It is usually applied as a clear coating. It forms a protective clear layer on metal. Lacquers are commonly used to prevent interaction between the container and the product. A wide range of coatings are used, including vinyl, epoxy, and phenolic resins. The aluminum tube can be coated on the internal surface with an epoxy resin based on lacquer to prevent reaction with sodium sulfite and also to prevent catalysis of oxidation, reaction in which sodium sulfite can be used up. Next slide, please. So the second solution to the problem is the degradation of the sodium sulfite follows pseudo second order kinetics. So what is pseudo second order? It is the reaction in which the substance normally follows first order kinetics. Due to the excess of concentration of reactant, it behaves in pseudo zero order. So as the reaction follows pseudo zero order kinetics, in this we can add overages. What are overages? Overages are excess amount of substances that are added in the formulation to maintain 100% of labeled claim during the manufacturing. This can be added to the 5 gram collapsible tubes, which can compensate the degradation of antioxidant so that recommended level of antioxidant is present at the end of its commercial life. Thank you, and now we are open for the questions.
Yes. So I have one question.
Yes.
I understand that you hypothesized with the mechanism of chemical degradation of sodium sulfide. Uh, I did not get the second part of it where you mentioned why it is more in 5 gram versus 10 gram in terms of the order of the reaction. Uh, when the problem statement says that it's the same product placed into two different packs, effectively meaning the concentration is same in both of the tubes. So how you would justify the order of the reaction being the reason of a difference between the degradation between 5 gram and 10 gram?
Uh, so there is a degradation, the means the constant degradation in both the tubes. There is the concentration is also the same, but as the amount in a 5 gram tube is lesser than in a 10 gram tube, so the drop is more significantly or easily can be seen in 5 gram tube. This is our means hypothesis.
Because we
That
generally measure assays in terms of percentage, not in terms of the absolute amounts. So from that angle, how you would justify degradation more in 5 grams?
the time it will time for 10 gram pack, it will be larger than the 5 gram pack if it is degrading at 10 mg per hour in both the pack in the both the packs, but in a 5 gram pack it will require lesser time than in a 10 gram pack. So the drop can be easily observed in 5 gram pack because it is in the time will be smaller or less time.
Okay, and any other hypothesis came during your discussions other than the one which you presented? Uh,
uh, I also think that it can be the volume of the tube so for the reaction to occur. Maybe the reactants will react in smaller volume more easily than in larger volume, because sodium sulfite will react more faster, and it will show the concentration drop more faster in smaller volume. That I felt about uh, during our discussion.
okay, yeah, yeah. Okay, just to understand from the coordinators, are the other participants as well there in the call right now who would be presenting next to this?
No, sir. No, sir.
No, sir.
All the other presenters are not here. Only the group
Okay,
who is presenting, that is only present. Rest all
great,
comes
great.
under the
Yeah.
organizers.
Yeah. So as a hint to the team, they should have dig further into the second option which you just mentioned on the volume of the tubes. So that would have got you closer to the probable most probable root cause in this case, I would say. Yeah, but thanks a lot. A nice presentation, I would say, and a nice attempt to reach to the the probable root cause. Thanks a lot. Thanks a lot.
Thank you,
Thank
sir.
you, everyone.
Thank you, sir.
Amazing work, team IDP 12. Thank you for
Thank
your
you.
time. Now I would like to call IDP 02.
Hello, I can't hear.
Can we
IDP2,
start?
you guys can start. Yes, IDP2, you guys can start.
Good afternoon, everyone. I'm representing IDP2.
There is a lot of backdrop noise in your side. Gargi, Dikshit?
Yes, ma'am.
Yeah, your voice is not coming clear because of this so much of backdrop noise.
Is it better now? Because I'm sitting in an isolated location.
अभी वो बंद हुआ था कुछ तो भी. Is there any kind of buzz going on continuous in the place where you are?
No, ma'am.
All right, ठीक है. So talk a little loudly so that we can understand what you're saying.
Okay, ma'am. I'm representing IDP2. Next slide. The problem statement given to us was an antipyretic molecule has been formulated as a powder for dispersion. This product contains high amount of sucrose for taste masking. The cost of product is quite high, and the major factor for the cost is the packaging material of the product, which is aluminium foil. There is a requirement from commercial team to reduce or eliminate sucrose as high content of sucrose limits their use in diabetic population. The team also requested to reduce the cost of the product. The objective was to formulate a product to meet the two requirements, that is taste masking and reduction of cost, without impacting end user experience. Next slide. Next slide.
The
Introduction.
slide has changed.
Sorry, ma'am.
Slide has changed. You can go on.
Antipyretic medications are used extensively right from children to elderly. Wide range of antipyretic analgesics are administered orally up to 4 gram in adults. Bitter taste of the drug makes oral administration difficult, hence palatability needs to be addressed. Taste masking should be done in accordance with the needs of the diabetic population. There are many research that address this issue like lipid coating or microencapsulation, but here we suggest only those which are both cost effective and also significant. Next slide. So for taste masking, either alternative sweeteners like sucralose, which is a non-calorie sweetener and 600 times more sweeter than sucrose, can be used. Hence, the quantity of the sweetener reduces, whereas sucralose and sucrose have almost similar cost. Or another method, that is alternative formulation methods, can be used. First being granulation. Granulation reduces the surface area of the bitter tasting drug, which comes in contact with the tongue on oral intake. Salt formation can also be used because solubility of drugs in saliva reduces, acting as an effective method for taste masking. And whereas in effervescent formulation, the medicament dissolves in bicarbonate water and efficiently masks the bitter taste of the drug. Next slide. For packaging material, we suggest two types of packaging materials, that is cellulose-based polymers for packaging and metallized PET film packs. Next slide. For cellulose-based polymers for packaging, cellulose is the plentiful biopolymer which has very low environmental impacts. There are several forms of cellulose, and the most commonly used for packaging is cellophane, which is a clear, thin, biodegradable plastic-like material. The environmental benefits of cellulose-based packaging is it is sustainable, biodegradable, and also compostable, whereas the packaging benefits is it is oil and moisture resistant, and the most important, it it is of very low cost. Next slide. The next packaging material we suggest is metallized PET film packs, that is polyethylene terephthalate film packs. In contrast to aluminium foils, which are made entirely of light metal, metallized PET foils are plastic foils which are only evaporated with aluminium powder in high vacuum. These films are made up of only 1% of the aluminium that would be needed for an aluminium foil. Hence, this pack is interesting as it is cost effective and also more environmental friendly as an alternative to aluminium. Next slide. The benefits of these packs are it gives a metallic shine and hence a special optical effect is seen. The aluminium vaporization improves its barrier effects against vapor, gases and aromas. It is a bit less gas tight than real aluminium foil, but it is more tear resistant, more flexible and lighter. Also, they are less sensitive to corrosion and not as wrinkle prone as aluminium foil. Next slide. To conclude, the cost of the product can be reduced by using cheaper alternatives for aluminium like cellulose or metallic PET sheets as packaging materials, and high contents of sucrose can be reduced by using less quantity of alternative sweeteners like sucralose or taste masking by different formulation techniques like granulation, salt formation, effervescent formulation. Next slide. These are the references and thank you.
Gargi, I would like you to switch on your camera for the Q&A.
It is on.
Somehow we cannot. Okay, now we can see you. Prabha ma'am, do you have any questions regarding this presentation?
Yeah, I have one or two questions. Gargi, good presentation. Okay, one question I want to ask you is you have said to replace sucrose with sucralose. Okay, what about the cost factor? Has that been come into account for you? Have you seen? And what is the replacement like? How much amount of sucralose needs to be added, which for in for क्या कह सकते हैं to overcome the amount of sucrose that is needed. So can you give any highlighted on that aspect of how you have put that point forward? That's good, but उसको justify थोड़ा सा if you could do it.
I checked for the cost of sucralose and sucrose. It is almost the same. That is, it was around 3,000 per kg for sucralose, whereas similar costing for sucrose was also there. And sucralose is more sweeter than sucrose, hence the quantity we can reduce. I can't see the exact quantity, but it would be definitely lesser than the amount of sucrose which is used for taste masking.
Okay. So then you would think it is cost effective as well as diabetic friendly, like that. Okay. And one more last question. You have used cellulose. You have shown an alternative as cellulose. Won't using cellulose polymers increase the cost of the things that you're saying? Packaging में cellulose कितना use होता है actually में? As a derivative, cellulose derivative. Do we use?
Food packaging nowadays, but it can also be used here because it is cost effective. Means I was checking the prices. Most of the articles they were claiming that it is cost effective, cellulose based packaging.
Somehow, and sir, what is your suggestion? Cellulose, is it most cost effective than aluminium? Because it's a sachet that is to be replaced with cellulose, you're saying.
Yeah.
Sachets would be much more.
Yeah, exactly. So my point was absolutely that. And how many products do you have seen in market with a cellulose sachet as such? You will not find a one, I believe. there are constraints, and when we say that end user experience, it includes everything. It includes the stability of the product. It includes cost is definitely commercial team will always ask for a cost, but you cannot compromise with the standard of the product or the quality of the product. So okay, so madam, you can continue if you have more question.
No, that's it. Those two questions were because we are looking at industrial problem ka industrial solution. So, it should make sense. I mean, even if you're using saying cellulose, uske फायदे जो है, you should actually then highlight that what would be the benefit be as compared to aluminium. Maybe something like that source. Because cost wise, I would not think.
More environmental friendly also, na, cellulose than aluminium.
One practical aspect I would like to give. It's generally, you cannot drastically reduce a cost by changing the materials. That's not generally, the. It depends. Like again, like you showed some with surlin coated or so that will add to some cost, but that will not be you will not have a drastic reduction with that. And let me know, because it might have come during your discussions or brainstorming. Did you thought of any other approach to reduce the cost? What was the prerequisite? Was the end user experience? It's not about the dosage form. Did you thought on that front as well in your discussions or brainstorming? So sucralose is agreed. Sucralose is one of the option where you can just reduce the amount of sugar significantly. So that's there, but that's part of the test. But what about the cost? Did you thought on something which would reduce the overall packaging cost? What is the least costly packaging? Why we see maximum number of products in that format, a tablet format or a that kind of blisters, for example? Did you think in that direction as well?
Yes, sir. We were thinking if we could change the dosage form only, but we were not sure if we could if we could suggest that as a solution. First thought we came across was why not a tablet and a dispersion, but we thought it might not be proper, so we didn't suggest that. We did come across this point.
Yeah. But if you see the wordings, it is nowhere says that we can't do that. It says that the end user experience remains the same, so it does not mean that you can't change the dosage form. Okay, but yeah, I would say a nice presentation and definitely provided some options. Even good part is you suggested multiple options of test masking as well, which I agree would need more optimization from a end user experience perspective. But yes, I agree. Those can be options going forward and reducing the amount of sugar. So nice work. Thanks.
Thank you.
Thank you, Gargi. I guess you can leave the meet. Next is IDP 8. I would request IDP 8.
Should we start?
Yeah, you guys can start. You guys have to keep your camera on throughout the presentation and Q&A session. Yeah.
Thank you. My teammates are joining me.
Yes, no problem.
Good afternoon, everyone. Our IDP code is IDP08, and the question given to us was, Can we move to the next slide? So the question was CSN capsule 2 mg, which is a hard gelatin capsule, was packed in PVC blister pack, and the product on 6 month stability showed was found to fail in the related substances. After evaluating the pre-formulation study results, it was found that the drug is sensitive to moisture. Also, there are about 3 to 4 months time which is available before planned exhibit batches. So the objective was what is the probable root cause and how to identify the right mitigation in given time without 6 months stability data. Can we move to the next slide? The causes. The causes that we identified were chemical stability, which was caused due to instability, which was caused due to oxidation and hydrolysis, and the moisture content of hard gelatin capsule, and packaging, which was done by PVC blister pack. And the solutions were so the solutions we found out were packaging material can be changed with PVC eclar, which is polychlorotrifluoroethylene, and as it has moisture barrier of PVC eclar is superior than the normal PVC. And we can also use aluminium foil, which is used for packing as it prevents moisture. Then instead of hard gelatin capsules, we can use HPMC capsules as the moisture content of HPMC capsule is relatively lower than the hard gelatin shells. Then we can use plasticizers, alternative plasticizers, which are not soluble in water. And examples are triphenyl phosphate, dimethyl phthalate. And we can also conduct accelerated stability studies for determining the oxidative stability.
Can we go to the problem statement, please? More time. Present that slide.
Yeah.
Okay. So my question is with respect to the last part of it. I got all the things with respect to the packaging change, but with respect to also there are about 3 to 4 months time available before planned exhibit batches. So how you would identify the right mitigation? If I understand you correctly, you would replace it with Aclar or maybe some other film in the blister. But how you would identify which one would be the right solution without having the 6 month data in this case, because you don't have 6 months basically.
Hello, sir. Sir, we can perform accelerated stability study for 3 months.
Accelerated at what condition? Because accelerated means accelerated 4075. Generally, we go for 6 month data to submit our data to the authorities. So how you would be confident going to exhibit batches with little or no lab data by that time?
Sir, in that case, we can. Yes, sir. So in that case, we can do is that we can perform matrixing of the containers that we have. So matrixing, which is in which our samples are tested at a particular specified time period. So 3 exhibit batches can be made, 2 pilot batch or 1 small scale, or 3 pilot batches. So 3 exhibit batches of different strengths or different packaging size container can be made. So as per FDA rules, it is stated that the accepted data of 3 months can be performed by use of matrixing. Yes, sir.
Yeah, I got it. And it's for exhibit what you are mentioning. My question is before that, because we are going for the exhibit and we would be putting in a X pack or a Y pack. Okay. So and if it fails at the end of the stability in exhibit, that's a huge cost to the company. So we don't want to do that. So how you would be confident going into exhibit, which one should be chosen in this case? and we have 3 to 4 months here.
Yes, sir. So exhibit data for two formulations can be made. So in this case, we can use PVC HPMC caps. We can also use different other types of caps as our different strengths. So that's how we can make. So that's how we can become confident about the product. So 3 months data of that product can satisfy our needs.
Okay. Okay. So just to probably provoke your thoughts and for future as well, there are tools available wherein you can actually evaluate stability in different packs without actually doing it in those packs. So we generally use modeling for that purpose. It's called as ASAP program, Accelerated Stability Assessment Plan, which you can Google and you can find more details on that. But yes, so it basically talks about studying your product at different temperatures, and it's not only 40, 75, 25, 60 or 30, 75, but more than that. The range would be bigger, till maybe even 70 degree as well, and with different ranges of humidity. We can run a statistical model on that with respect to the degradation. And then there is a, so that model will give you some at least some conclusion on how your product will fare when a X amount of MVTR is there. So you know MVTR of each of these materials, so then you can actually select the right material for your product. So that's the way you could do that. The other parts I think were quite clear. So I agree with that solution that you would definitely go to a higher grade material, in this case, for example, Acrar, because that will provide you better protection from moisture. So that's I think appropriate solution in this case. Thanks a lot. So that was last question from my side. Open for others for the questions.
Good presentation, guys. I just would have liked you to give a little bit more, solutions. I mean, you just, whenever you're giving solutions, there has to be a lot of justification, at least some amount of justification in the given time. I understand, but still, I felt that there is very little justification and just statements kept there. Okay, this is what we can do, why we can do it, what is the accessibility. Bray, you put 2 points, but explain those 2 points to make sense of your suggestions. Okay, so just next time, just keep that in mind. Okay, do not make too many slides, but at least while presenting, thoda sa or detail mein you to take only 2 good solutions and put that, rather than giving 4 or 5 solutions and not being able to justify or explain them properly. That makes no sense. Okay, so that is one of my suggestions. Okay, okay, good.
Thank you, guys. You guys can leave the meet. Now, I would request IDP 14. Guys, you all have to switch on your cameras during the presentation and during the Q&A session.
Just a second.
Yeah. Take your time.
Are we all visible?
Okay, how many students are there? 4? Okay.
Yes. So good afternoon, good evening, everyone present. So I'll be presenting my slide. I can't see my slide. Okay. So good evening, everyone present here. So the code given to our team is IDB 14. So the problem statement given to us is an antibiotic molecule has been formulated as a powder for dispersion. So the unit weight is 8 gram and the drug content is 500 mg. So this powder for dispersion is dispersed in 150 to 200 ml of water. The product contains high amount of sucrose, which isn't suitable for the diabetic patients and is used for taste masking. And
The major part of the cost is aluminum foil sachet, and the commercial team wants to reduce or eliminate sucrose as the high amount of sucrose is not suitable for the diabetic population. Next slide, please. Next slide. Previous.
1 second.
We are not able to see screen.
The presentation
Just a
is
minute.
not.
Just a minute.
Okay. So the objectives extracted from the statement are to reduce or eliminate sucrose as high content of sucrose limits usage in diabetic population. Second, to reduce the cost of the product. The major part of the cost of the product is the packaging material, which is aluminum foil sachet, and we are told to suggest ways to formulate the product to meet above requirements without impacting the user experience. So the solution we have given is, next slide, please. So instead of using sucrose as taste masking agent, we can use sucralose, which is an artificial sugar, and it is 800 times sweeter than sucrose. So sucrose is suitable for the diabetic patients, and it will reduce the weight of the final formulation, and thus reducing the uh, size of the package, that is the aluminum foil sachet, and thus reduce the uh, cost. And instead of sucrose, we can use hydroxypropyl beta cyclodextrin and complex it with the drug, and it can be used as it has an advantage of taste masking, and it will also help in the dispersion of the powder. Next slide, please. And also we can change the formulation technique. So instead of formulating powder for dispersion, we can formulate dispersion tablet. So in this the tablets can be packaged into a blister packaging. So as we know, instead of 10 aluminum foil sachet, only one blister packaging will be used, so the cost can be reduced up to 10 times. And this tablet can be dissolved in warm water and can be drank. Next slide, please. So as you can see, this is the graphical representation. So the drug can be of BCS class 1 or class 2. For BCS class 2, the hydroxypropyl beta cyclodextrin can be used for the taste masking, and it will also help in the dispersion of the tablet. And for the taste, we can add slight flavor. And in the case of BCS class 1 drug, we can add, instead of sucrose, we can add sucralose. So it will reduce the amount of excipients, and for the dispersion, we can add super disintegrant and directly compressible excipients. So the dispersible tablet will be formed. Next slide, please. Next slide. So these are the formulation tables uh, for the BCS class 2 and BCS class 1 drug. So in the case of BCS class 2 drug, the complexic agent will be used in the concentration of 1:1 ratio, and the super disintegrant will be used in the concentration of 5%, and the total weight will go up to 1245 mg. And in the case of BCS class 1, we can replace hydroxypropyl beta cyclodextrin with the artificial sweetener, that is sucralose, which is used in the quantity of 2%, and it will reduce the total weight of the tablet approximately to half, that is 715 mg. Next slide, please. Next slide. So for the summary of the solutions for the objectives, for the objective 1, if we don't want to change the formulation technique, we can use artificial sweeteners instead of sucrose, which will decrease the amount of final formulation and thus reducing the uh, size of the sachet, which will in turn decrease the cost. And instead of sucrose, that is the sugar, we can use the complexing agent hydroxypropyl beta cyclodextrin as taste masking agent, and it will also enhance the dispersibility of the wetting of the powder. Hence it will decrease the dispersing time. And for the objective 2, aluminum foil sachet can be replaced with the blister packaging if the dissolving tablet is formed instead of granules. And in one blister packaging, 10 bt tablets will be can be incorporated, and the cost will be reduced up to 10 times. Thank you.
Okay, Tarab, I have one question for you.
Yes,
beta cyclodextrin complex. So isn't I mean, you're ek taraf you're talking about cost, and ek taraf beta cyclodextrin you're saying. Don't you think it is going to majorly increase the cost? You know that it's always a problem of bonding, and would tub अंदर लेना, and what is the molecular weight? Have you considered you know that one molecule one, so it is this attachment by itself practically देखा जाए तो it is very expensive, right?
Yes, ma'am.
I mean, we do, of course, as researchers, we always do beta cyclodextrin complex and we enhance solubility. That is theoretically, but practically will it make sense, especially with the BCS class 2 or class 1 drug with high molecular weight. I mean, यहाँ पे तो कोई parameter दिया नहीं है, so you're just assuming. Yes. So fine. Okay, so that थोड़ा सा it does not make sense. And in taste, how is beta cyclodextrin used for taste masking over here? Can you tell me?
It encapsulates the drug, so the taste masking is an advantage of it.
You are enhancing solubility of a drug by using beta cyclodextrin generally, and then you're saying कि taste mask भी हो रहा है, right? So, yes, but don't you think when the solubility of a drug gets enhanced, because it is a beta cyclodextrin, so then the bitter taste of the drug will be much more prominent? How is that going to help in encapsulation? And then, I mean, if you would have said ion exchange resin, I can still understand, because there actually that will happen, right? But how does beta cyclodextrin help in that? That I could not understand the logic. Yes, Darshana will answer for us.
It encapsulate the drug.
Why use beta cyclodextrin as an example for encapsulation? If you simply want taste masking, you can use so many other normal neutral polymers, as we call them, without having beta cyclodextrin, which is in regards enhancing the solubility of the drug. The more soluble the drug, and if it is a bitter drug, obviously the more taste it's going to give. I mean, it is beta cyclodextrin is not meant for. I mean, you must have thought it's, but it's not meant for taste masking, isn't it? No, you can counteract and tell me if it is. You must have seen it in somewhere in literature. Has it been mentioned? Something like that. So you can give an answer.
Yes, ma'am.
Yes, what?
And we found in literature that it can be used for taste masking if the drug is getting encapsulating inside the beta cyclodextrin.
And was it the first option that they have shown that can work effectively?
Not as a first option, but as an alternative.
So before even you think about, so as researchers, you all are researchers, right? So as researchers, I mean, when we think about it, okay, so it's like whenever you have to taste mask, okay, what is the first thing that you go for? It is not cyclodextrin that you will go, okay, let's go to cyclodextrin to taste mask. So see, because it is not going to, it's very expensive. Second, इसका docking होगा नहीं होगा, who knows? That is is a different art, what you can say, analysis altogether to get that thing done. So I'm just saying ki
Our
your
major
good
solution is
alternative,
using of
but you
pseudo.
suggested an alternative. Is it the best alternative that you could find? Is my question. It is an alternative, but is this the best you could do?
No, ma'am.
हाँ, because you all the इतना तो literature search किया ही होगा ना?
Yes, ma'am.
Okay, so it's not a
So our
bad
first
answer.
option was
Okay, I'm not saying
sucralose.
it's a bad answer,
Yes,
but it could have been better.
yes.
है ना? है ना, Jayesh? It looks like Jayesh is going to sleep, sir.
Oh man.
Akka. He's echoing for us. Okay, ठीक है. All right, sir, you can go ahead and ask question.
Yes, so my question was that. So yes, because and this is a very classic example where what's the basic objective of this whole exercise is actually, once you read the problem, so we need to see a bigger picture. It should not be like just start thinking on something which can be a research concept basically. So when we read that as a problem, you can get a sense that what type of product it is. Okay, so if you see the product looks like more of a over the counter product. So it's an antipyretic. It's a powder for dispersion put into 200 ml of hot water. So all those points towards something which is routinely used by people, and it's a OTC product where the cost is very important. Okay, so in those cases, we never go for something which is very fancy and without knowing it's really important, it's useful or not. Okay. First part is that. Second is, when we say that its end user experience should not change, which includes, and Madam rightly pointed out that you are going to put that complex into a 200 ml of hot water, you're actually improving the solubility, and then you are expecting that it would not be bitter. So that's something may not be the first option for test masking in this case. There may be very very simple physical options available wherein you can just coat the drug particles with something which will act as a barrier but may still impact the user experience, giving that gritty feeling into the mouth. So those kind of things which you need to imagine. It's not always Google which may help every time. So it's more of an imagination and looking at the big picture. But I must congratulate you that you thought in terms of the second part, you thought about the changing the dosage form of the product. That's most effective solution, I would say, in this case, to reduce the cost, to put a sachet into a tablet. Okay, so tablet is a fast manufactured dosage form. It can be packed very easily without much of issues. And again, if it's a dispersible tablet, would never change the user experience. So that's a very good thought in the or the way it was put. I really like that. So I must congratulate for that. The first part, yes, sucralose is the option. You could have provided more options to test mask it. There are multiple, actually. There are multiple, and beta cyclodextrin is really probably last of the list in this case. So yes, so one part is very good. The other part, I would say, you could have dwelled more on other options in test masking. But yeah, in all, presentation wise, it's good. Yeah.
Yeah. More scientific. So that's good to see.
Yeah.
Thank you, sir.
Thank you,
ma'am.
Thank you.
Okay.
Thank you, guys. IDP 14, you guys can leave. Our next.
Yeah, I just want to say, sir, I'm just going to the office, so I'll be back in 10 minutes.
Ma'am, should I wait for you?
Okay, madam. Okay, no problem.
No, continue. Please continue. I'll just go to office.
Okay, madam. All right.
Sir, should I continue?
Yes, you can continue.
Okay. Our next IDP is IDP 03. Guys, you guys need to keep on your cameras on throughout the presentation and Q&A session, please.
Shall we start?
Yes, guys, you guys can start.
Good afternoon, everyone. Our team code is IDP03. Our problem statement is a nasal decongestant spray formulation is being developed. During development, it was observed that the formulation residence time in the nasal cavity is not sufficient to produce its pharmacological action. So our objective is to suggest improvements in the nasal spray formulation to increase the residence time in the nasal cavity. Next slide, please. Next, so first let us understand the anatomy of the nose. Our respiratory system consists of the upper and lower respiratory system. Nasal cavity is a part of upper respiratory system where it consists of nasal vestibule, respiratory region and olfactory region. Next slide, next slide. So here mucociliary clearance mechanism is naturally present where it is to prevent any entry from the foreign particle into the respiratory tract. Cilia, which lines the nasal area, beats in such a coordinated fashion that it propels the foreign particle trapped in the mucus towards the nasal opening, which is later coughed out. This mucociliary mechanism highly affects the absorption profile of the nasal drug because the drug residence time is reduced by this clearance mechanism. Next slide. Here, the nasal congestion is a pathological condition of a nose. Here, mucosal inflammation leads to problem in breathing. It is present in various diseases. The mucosal inflammation is also responsible for other interrelated factors that contribute to the nasal congestion, which are venous engorgement, tissue edema, and increased mucosal secretion. Next slide. Here the nasal decongestants comes into picture as they are used to relieve nose congestion. It is used in various diseases such as hay fever, rhinitis, common cold, etc. It should be noted that it gives short-term symptomatic relief and does not cure the underlying condition. Next slide. Nasal decongestants belong to the alpha-adrenergic agonist class. The site of action is the pre-capillary and post-capillary blood vessels of the nasal mucosa. It acts by constriction the blood vessels which reduces the supply of blood, which leads to the reduction of mucosal edema. This is the targeted area of the nasal decongestants. Next slide, please. These are some marketed formulations for the nasal condition, nasal decongestant. Next slide.
The reasons we choose intranasal route for administration are because they are non-invasive, easy to administer, onset of action is rapid, higher bioavailability of the drug as compared to the oral route. It also serves as an alternative for rapid delivery of the drug when intravenous route is not accessible. The disadvantages of the intranasal route are the drugs which are highly lipophilic in nature cannot be administered easily. Small molecular weight medicaments can be administered. Drugs which do not lie in the pH range of 5 to 7 may cause irritation to the nasal mucosa. Bleeding, congestion may also hinder the administration of the drug. Next, please. Nasal decongestants relieves congestion in the nasal tissue caused due to swelling of the blood vessels, for example, oxymetazoline hydrochloride, phenylephrine hydrochloride. Accurate doses can be administered, that is 25 to 200 microliters per spray. Dose accuracy is influenced by thixotropy, surface tension, viscosity, orifice size, force, design of the pump of the formulation. Next slide. Next slide. The limitations. The volume of the drug administered per spray is limited, that is between 25 to 200 microliters. Therefore, only potent drugs can be administered. Molecular weight of the active ingredient must be less than 1 kilodalton. Small area is available for absorption. Contact time of the drug at the nasal epithelial is less due to the presence of mucus. Mucociliary clearance is also one of the limitations. Next slide.
The residence time of a nasal formulation is time for which the drug formulation is retained in the nasal cavity. We can improve the residence time of the nasal formulations by applying the following strategies, which is to add the viscosity enhancers, bioadhesive excipients, absorption enhancers, and using dry powder formulations instead of liquid. Next slide. Viscosity enhancers like HPMC and methyl cellulose increase the viscosity. This retards the diffusion of drug from matrix and causes retention of drug in nasal cavity, which increases the therapeutic efficacy. Next slide. The mucoadhesion is mechanism when excipients interact with biological membranes through different interfacial forces and are retained for prolonged periods of time. Mucoadhesion takes place by wetting followed by establishment of adhesive interaction. This controls the rate of extent of drug release, which increases the frequency of drug administration, which decreases the frequency of drug administration and improves the patient compliance. Next slide. Some of the bioadhesive excipients are microspheres, liposomes, and gels. The nasal formulation absorbs water, resulting in swelling and formulation of gel layer. This gel layer will be in contact with nasal mucosa and result in rapid absorption. This will increase the residence time and cause high bioavailability. Next slide. Some of the bioadhesive polymers employed are carbopol, sodium hyaluronate, guar gum, and the mucoadhesive force of polymer depends upon its nature, the pH of surrounding medium, swelling, and physiological factors such as mucin turnover and disease state. Next. Gels. Inclusion of gels increases the viscosity, which reduces post-nasal drip and taste due to less swallowing. The anterior leakage of the formulation is also decreased. It is also less irritating and better for targeted delivery to mucosa for better absorption. Next. Microspheres are usually based in mucoadhesive polymers that are chitosan and algin. Microspheres protect the drug from enzymatic metabolism. One such example is a degradable starch microsphere, which is Ferex. Next. Liposomes are bilayered phospholipid vesicles which contain one or more aqueous drug compartments. Liposomal drug delivery presents various advantages such as encapsulation of small and large molecules with a wide range of hydrophilicity and pK value. This increases the retention time by protection of entrapped drug from enzymatic degradation and mucosal membrane disruption. Next.
Next. Addition of absorption enhancers increases membrane permeation rather than increasing solubility. So they are also known as permeation enhancers. Now there are four mechanisms of absorptions: transcellular diffusion, paracellular transport, vesicle mediated transport. The mechanism of absorption enhancer is to increase the rate by altering the structure of epithelial mucosa. This should not cause any damage to the mucosa or permanent change to it. Increase in residence time does not always necessarily lead to increase in absorption because the agent increases the residence time but not necessarily promote absorption. This leads to less viability of the drug at the site of action. Therefore, an absorption enhancer is required. Next. There are four mechanisms of absorption enhancers: open tight junctions, inhibit enzyme activity, decrease mucociliary clearance, and reduce mucous viscosity or elasticity. The ideal penetration enhancer should have the following characteristics. It should carry the drug molecule across the cell from apical to basolateral surface. The carrier should be potent, pharmacological inert at the concentration used, and cause no irritation. It should be compatible with the drugs. Next. Types of absorption enhancers: surfactants, fatty acids, cyclodextrins, phospholipids, bile salts, chelating agents, glycols, prodrugs, co-solvents, enzymatic inhibitors. Surfactants are the most effective permeation enhancers, but because they cause epithelial toxicity and nasal irritation, they are not preferred. Cyclodextrins such as alpha and beta cyclodextrins, methylated derivatives of them, are proven to be the best soluble absorption enhancers. Enzymatic inhibitors
Nasal mucosa is an active enzymatic barrier as it contains various metabolizing enzymes. So we use proteases and peptidases in the formulation of protein and peptide. Use of bile salts also avoid enzymatic degradation. Bile salts are found to be less irritating to nasal mucosa, decrease in hemolytic activity, and decrease
Guys,
in
your time is almost up.
decrease in protein release as compared to surfactants. It also affects the nasal membrane by creating temporal pores and enhance the permeability. Next,
Guys, you
these
guys
are the
only have 30
examples.
seconds.
These are the examples of the absorption absorption enhancers. Next,
Go to the summary slide now.
use of dry powder formulations. One second. Use of dry powder formulations. The use of dry powder formulation instead of liquid spray results in better deposition and residence time.
Guys, you
These
need to stop.
formulation.
The time is up.
Okay. Thank you.
Okay, I think there was a lot of matter in the presentation. Okay, as a team, which one would be the best possible solution for this issue?
So including all the various excipients in proper proportions will be a better solution for this problem, because sometimes the dry powder formulations may cause irritation and may not be patient compliant. So addition of viscosity enhancers and bioadhesives would be a good solution to this problem.
Okay, so if you go back to the problem statement, I think the problem statement clearly says that it's a spray formulation which has got a very little residence time. So you have to include the residence time. It's my just to understand basically why you focus more on absorption enhancers.
So like seeing our problem statement, it says that we have to increase the residence time of the formulation which is developed, formulation of nasal spray which is being developed. So we have focused on absorption enhancers because it in the literature it says that the agents which increase the residence time, uh, it may increase the residence time, but it is not promised that the absorption of the drug is like for sure in the nasal cavity. Therefore, absorption enhancers are required in the formulation.
Assuming the absorption is not good in this case, basically.
Assuming the
Right?
absorption is
Not great, basically, because why you included, because you thought that this particular drug will not get absorbed from the nasal cavity quickly. Yeah.
Yes, sir.
Yes.
Okay. There was one more solution which you presented in terms of viscosity. So what would be the challenge of increasing viscosity into this formulation, and what can be the solution for that?
As it is a nasal spray, so if we increase the viscosity and the retention time is more, it may not be patient compliant because the patient may feel that there is something in the nose and breathing might also be a trouble. So the viscosity enhancement should be adjusted to an optimum level, as in it must not flow away when it is sprayed, and it must not be that viscous that the nostrils will stick, which will impair the breathing.
Any other problem you anticipate with increasing the viscosity other than that feeling of the an unusual feeling? Other than the unusual feeling, what may be other problem? Maybe a very practical problem.
The spray formulation, as sometimes it is in form of an aerosol. So if there is increase in viscosity, the formulation may not come out of the container properly. So that may be a problem.
Okay. Okay. So have you, whenever you you must have done a lot of literature search for this particular problem. Did you also look at what products we have in India for this type of decongestion in market? Are those aerosols, or how are those basically? Spray
So there
formulations, I
are
would say, not the tablets. Yeah, you showed some tablets as well in the presentation, but yeah, the I'm interested in spray formulations.
So we gave the example of a holistic marketed formulations. Like there are various nasal decongestant in the form of tablets, capsule, and nasal sprays. So for the particularly for the nasal spray, there is a marketed formulation called Otrivin, which consists oxymetazoline as the main API agent. And like considering all the, we have just considered all the nasal decongestants, but however our focus is on the nasal spray formulation.
So just one suggestion maybe, and it's not a question actually, it's a suggestion. Then when you are working on a problem statement, be very specific, because you could have avoided slides in terms of how the ciliary mechanism works and all those things, because that's not our problem. That's more of a theory. It's more of a review article on how you are going to do a nasal delivery. So that is what not in this question. The question was very specific, that how you would improve the residence time. I agree with few of the points which you presented. One is viscosity. Definitely, viscosity increase would increase the residence time. I'm not very aligned with incorporating absorption enhancer, because that is what is not required. It is more of assumed in this case. It depends on molecule many a time, and many molecules work locally, don't require at such a big absorption, and they still work. Okay, so and you could have gone more detail into and it's more of an example. So you can just pick some marketed product to dig more on those molecules and see how they really work. So probably you may get an answer that do we really need a absorption enhancer in this case, this particular problem. With this viscosity, I think the one problem generally comes up with the spray formulations is the sprayability of that particular formulation. So as you go on increasing the viscosity, it never comes out of the bottle at the end. So that's a very critical and physical problem which is presented, and that needs to be solved. So do you can offhand think of any option to improve that? I just want a good viscosity. At the same time, I just want a spray as well.
Sir, we can, instead of spray, we can use drops if we need more viscosity. So it will give appropriate amount of dosage at the time.
Yeah.
So and
Just a
in
hint.
addition to what my team, okay, sorry.
Yeah, go ahead. Go ahead. Sir. Go ahead. Go ahead.
So and in addition to what my teammate said, we can also use some carriers instead of increasing the viscosity viscosifying agent. There are some carrier agents like cyclodextrins, which can be used to like deliver the API like without any hassle. So, like the complexing agent, which is cyclodextrin, that can be also used in the formulation.
Okay. Yes, one more hint I probably would give, and you can search later. It's not like for this presentation, but definitely you can think on. Is there, we can search for polymers which can actually give a viscous dispersion, which is thixotropic, which changes the viscosity with the mechanical force. Okay, so there are polymers which will which will lower viscosity when you apply mechanical force like spraying or spray pump, and they attain viscosity back, or they, then you can also think about something which is inside to gelling into the nasal cavity, wherein you spray it as a suspension, and it will gel inside the inside the nasal cavity, and would stick over there for longer period of time. Those will be more practical and easier solutions as compared to adding a microsphere or a liposome, which will take longer period of time to develop, stabilize, and then deliver ultimately. So it's a simple solution, spray formulation. We don't want to add more complexity. When you say, I will add a absorption enhancer, actually you are going towards, making this more complex. You would have to think about what is the bioavailability. You will have to go into, bio studies to show that, my earlier versus this formulation actually exactly gives same bioavailability. Ultimately, it's not so power. So those kind of consideration also comes into picture. So, but okay, nice presentation. Number of options you gave, and that's why I asked which one is the favorite of favorite in this case. But yes, thanks a lot. Thanks a lot. Nice presentation.
Yes, one general question I would like to ask. Sripas sir covered all the technical aspects, but I am more interested about collaborative efforts, whatever you had taken to come out with these presentations. Maybe solution will be good, better, or best. And Sripas sir justified that thing. But I would like to ask you, after getting this problem statement, in what way your thought process got evolved and you came up with these presentations collaboratively? Would you like to add anything on this?
We learned more about nasal drug delivery system because of this project, and we learned that how different NDDS novel drug delivery systems can be used to increase the retention time or absorption of the drug into the body. Uh, so basically, we learn more about every excipient and novel drug delivery.
Yeah, my
Sorry.
question is about collaborative efforts. In what way you came up with the presentation? Means what was the thought process? In what way you collected or collated all the data and presented?
So we actually thought of we searched about what is the problem. Like we got the problem statement and then we searched about the complexity of the nasal drug delivery system. Then we started, collecting the information from various review articles and everything, like how the formulation has been done and how they have improved it. So we were searching for that point only. That's why we came up with viscosity and bioadhesive, mucoadhesive gels, liposomes, and absorption enhancers. That's why we put it in the last. Absorption enhancers was not particularly, but it was giving us some hint because there was a controversial statement about it. Even if we increase the residence time, but the absorption is not increased. That's why we kept it in the last. So it was a controversial statement because we wanted to be sure about it. We did not want to miss anything in that. That's why. And it was that way.
Okay, all right. A nice presentation.
Yes.
Yeah.
Yes, sir. Thank you, sir.
Okay. Bye.
Thank you, IDP03. You guys
Thank you.
can leave. Next is IDP05. IDP05, you guys need to keep your camera on throughout the presentation and during the Q&A session. You guys ready?
Actually, hello, I'm not able to on my camera.
Okay. Other team members, you guys ready?
Yes, am I audible?
Yeah. You guys are audible. Okay, you guys can start then.
Okay.
So good evening everyone. We are team IDP05 and we are here to present a solution regarding the given problem statement. Innovator's patent has an independent claim as product containing not less than 98% crystalline API, and we have to develop a non-infringing formulation. Next slide. So this statement suggests that the product must contain at least 98% crystalline API. The problem in developing such formulation is that there is a possibility of the API losing its crystallinity, and the crystals also have the ability to exist as one or more polymorphs. The crystalline state of API will definitely affect its solubility. Solid form modifications such as solvates or hydrates, metastatic polymorphs, and salt formation are some approaches to improve the physicochemical properties of the API, but they all have their individual disadvantages. Next slide. So to overcome all these issues, we have found a best approach, that is co-crystal based formulation. Next slide. Next slide. Yes. So co-crystals are multi-component systems containing an API and a co-former in the same crystal lattice. Here, the crystallinity of the API is maintained throughout, and the co-former is responsible for changing its physicochemical properties. Next slide. Co-crystal solid form provides these advantages, which include dissolution, stability, solubility, taste masking, controlled release, multi-drug co-crystals, bioavailability, and patentability. Next. Co-crystals are the novel solid forms of an API, and they have significant industrial utility, and its synthesis involves non-obviousness. Therefore, co-crystals are patentable inventions as they satisfy all the three eligibility criteria for novelty. Those are industrial applicability, non-obviousness, and novel structure. Next slide. Co-crystallization is a novel technique to enhance drug solubility, dissolution, and bioavailability. Industrial production of co-crystals are scalable, robust, and environmentally friendly, and techniques like spray congealing do not involve use of any solvents. Thus, it is a green technology. Some of the commercially available co-crystals in the market are Suglat, Entresto, and Stigletro.
Next slide, please. Co-crystal production involves two methods, solid state methods and solution state methods. Next slide, please. Next slide, please. Solution state co-crystallization offers a number of advantages over solid state crystallization. It also assures the purity of the co-crystals. Next slide, please. Solution-based techniques involve slurry method, solvent evaporation, anti-solvent method, and supercritical fluid method. Next slide, please. Now coming to the challenges expected in developing this formulation. Next slide. First, solubility and thermodynamic stability. So the selection of co-crystal having optimal solubility remains a challenge, as distinct solubility behavior may be observed under different conditions of pH, temperature, and presence of solubilizing agents like lipids, polymers, surfactants, etc. Thus, adding appropriate pH modifying agents like phosphate buffer solution, surfactants like 4-hydroxybenzoic acid, polymers like polyvinyl and pyrrolidine can help increase solubility of co-crystal. But use of these agents needs to be in a proper stoichiometric ratio, which is necessary for its solubility. Measurement of eutectic constant is valuable in evaluating the effect of solubilizing agents and pH on solubility and thermodynamic stability of co-crystal. Second, dissolution. So particle size reduction increases the surface area, which results in increased dissolution. Co-crystallization of a drug may also increase dissolution rate. Faster dissolution of co-crystals reduces time to absorb drugs from the gastrointestinal tract, which eventually minimize the effect of food on oral absorption. Third, microenvironment pH. This affects solubility and stability. So for example, in acidic microenvironment pH, co-crystals of adifovir dipivoxyl AD with saccharin were more stable and soluble than co-crystals of AD with nicotinic acid. So this is because the basic environment created by the nicotinic acid co-former, which triggered the degradation of the drug adiphovyl dipivoxyl. Thus, it is necessary to maintain microenvironment pH by adding suitable buffers. Next slide.
Next slide. Yeah. So the fourth one is permeability. The permeability is major barrier in achieving the desired absorption and distribution of the drug through biological membranes. Addition of permeation enhancers disrupt the normal integrity of these biological membranes. However, co-crystals have opened new avenue to modulate permeability of drugs, without modifying the covalent, adopting covalent modification. So thus permeation enhancers such as EDTA can be incorporated in these formulations. The next one is chemical stability. So the importance of selecting right conformers based on the degradation pathway of a drug to prevent its chemical degradation has been recently illustrated in the case of adiphovyl dipivoxyl, that is AD. Co-crystals of AD formed with a weakly acidic coformer, that is saccharin, exhibited a significantly greater chemical stability than the co-crystals formed using the basic coformer, that is nicotinamide. This varying behavior of the co-crystals of AD was attributed to differences in microenvironmental pH. Therefore, appropriate buffers are needed to be added. So, the next one is physical stability. Several studies have demonstrated that stability of drugs towards moisture can be improved, deteriorated, or remain unaltered on co-crystallization. Therefore, to avoid these problems altogether, desiccants can be used to reduce moisture content. Right choice of container closure system can help in the protection of co-crystals products from oxygen, moisture, and light. Co-crystals of THP with acidic coformers such as malic, or glutaric acids undergo dissociation in presence of water. Therefore, control measures such as manufacturing in low humidity condition, maintaining the drying temperature below the transition temperature would help in tackling these problems. Drug excipient compatibility is also an important part. For example, dibasic calcium phosphate dihydrate is known to undergo hydrolysis during to release uh, to release water that may trigger the dissociation of co-crystals, leading to physical instability. Thus, beyond the traditional drug excipient compatibility study, compatibility of co-crystal with excipients should be performed in development of these drug products. So that's all from our side. Thank you.
Thank you.
So, yeah. So my question is basically, what did you understood from the problem statement? Can we just go back to the problem statement? Right. Okay. What was the understanding? Because if I am getting the sense that the understanding was different than what I understood from this problem. So can anybody explain what is the problem here?
Okay. So just a second. So we got the statement that product containing not less than 98% crystalline API. That means the API is in crystalline form. For 98% of API is in crystalline form. And what was expected is a preferred approach to develop a non-infringing formulation. That means we must not disobey the statement. Like the product, whatever
Yeah.
product.
Can you interrupt? Sorry.
Okay.
So when we say non-infringing, okay, what's the team's understanding on the word non-infringing? It says that innovator patent has an independent claim saying that product containing not less than 98% crystalline API. So what innovator is actually claiming is that in his product it's 98% crystalline API. So if I want to make a non-infringing product out of this particular, coming out of this particular patent, what would be my preferred approach? Yeah, I'm not talking about, a co-crystal or any other approach. I'm just talking about, from understanding point of view, what would be the basic requirement in my product, not the innovator, because innovator is already there. Okay.
So since it states that it's non-infringing formulation, so my product
Yes.
must contain 98% crystalline API.
Then it would infringe the patent of the innovator. The patent says that it contains 98% crystalline API, and the innovator will sue you saying that, okay, you are just copying my product.
But actually, infringing is an act of not complying with the patentee statement, like what the patentee has suggested here, that product containing not less than 98% crystalline API. If I may, infringing that statement would be like I'm not complying with that statement, or making a product completely not having 98% of crystalline API. That means I'll become an infringer, and the patentee can sue me for the infringement.
No. Okay. So let me just, I don't know, madam already has a background of IPR, so she can add more to this. But uh, uh, see, whenever we say a non-infringing means, we are not falling into the, independent claims of the innovator or the patent holder. Okay. So if the patent holder says that, his product contains 98% of the crystalline API, that means we need to have less than 98% of the API in the crystalline form actually. So here it's not required. Actually, it's infringing when you actually copy the innovator. Okay. So you don't have to copy. Actually, you have to go away from whatever the innovator has claimed in his independent claims. Okay. So a non-infringing thing is, say for example, I will give a simple example to explain in this case. Say for example, a innovator says that uh, uh, I have a water bottle, made up of PET, okay, PET, okay. So a non-infringing product would be a water bottle of a metal, okay. So you go away as far as possible from that particular claim. Okay. So if the claim says that it's PET, if I use any other polymer or if I use any other material, I actually don't infringe that patent. If I just use PET, that means I'm copying the innovator, and then innovator sues you saying that, okay, you just copied my innovation, you just used a PET, so you are actually copying and you pay royalty, or I will sue you. Okay. So that's what a non-infringing. So I think you got it somewhat differently, and that's why you came up with a co-crystal theory. In that case, suppose now if that is clear to the team, what would be the approach in this case to formulate a formulation?
Then we'll not stick to crystallinity. We can convert into amorphous state of the drug. So it can, like for solid modifications like salt formation or solvates, like these can be the approaches, wherein it actually converts into amorphous form of the drug, and even the physiochemical properties can also be increased, like poor solubility or dissolution, bioavailability, permeability, etc.
Okay. What and again, second part of the problems statement was, what would be the challenges with that approach? Okay. So what would be the challenge if you go with some other approach, like a solid dispersion, for example, or converting into amorphous form, or converting into a solvate, or converting it into a even as you rightly mentioned, co-crystals, there are co-amorphous material as well. Okay, so you can go to that. But what would be the challenges if you really go on that side? All of these, I'm not speaking about a particular thing, but as a general, what would be the challenge when you go away from a crystallinity of a molecule?
Uh, Since it's an amorphous state, I guess it will be unstable. It has a possibility to return to its crystal structure. So we must maintain that amorphous state. And also in any like salt formation, again, if I'll take salt formation, again, it depends on the acidic or basic functional groups present on the drugs. Like it targets the acidic and basic functional group of the drugs. And amorphous is soluble, so solubility is not an issue. I guess crystallinity,
Can it
it
be an
can
issue? Can it be an issue? Solubility can be an issue. Increased solubility can be an issue.
increased solubility.
Is it always required to increase solubility for all the molecules?
No. Amorphous, for
So
amorphous,
what
increased solubility
yeah,
is not an issue.
but what would be a problem with increased solubility?
Increased solubility can also cause increase in the excretion of the drug faster from the body.
Yeah, it can also, yeah, you reach to the last part of it, but what about the first part of it? Increase the absorption of the drug? Going into, not similar to innovator, because, it would be absorbed quickly as compared to the crystalline API, and it would, have a very, very high Cmax, very low Tmax. Uh, 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 would that be a problem? You one problem, yes, you rightly said, the stability of the amorphous form itself. So that's one of the key issue always with the amorphous materials. So that's one. Second is the solubility, because increase in the solubility many times is not desirable, because the innovator is a crystalline innovator. So when the innovator is crystalline, it already proven bioavailability with a crystalline form. You are going back to amorphous, you probably will end up into a supra available drug, which would not be the desired outcome, because you need to match exactly the uh, bioavailability of that product with the AUC and Cmax and Tmax as well. So yes, that was the thought process actually. And if you see, if you look at the infringing and non-infringing, I think that was the major miss in this case. I would say, for the presentation from other aspects, it was a good presentation, I would say. But yes, definitely you can dig more on what's infringing and non-infringing, and how you think about. See, you cannot copy what is already there in the independent claim. So you need to come up with it, which is unique, novel, different from what is already claimed. Okay, so and that makes it non-infringing basically. So good, but nice try. Thank you. Yeah, and it's open for others to ask questions maybe. So if not, I think I'm done with my question.
Thank you, sir.
IDP05, thank you so much, guys, for your presentation. It was amazing. You guys can leave the meet.
Thank you, sir.
Thank you.
I would now like to have IDP13.
How many more presentations, Heta?
Sir, there are 3 more, I guess.
Okay.
Sir, 3 more after this. After IDP13, there are
This
3
is-
more.
Okay, all right.
Yes, sir. IDP13, guys, you all have to keep your cameras on throughout the presentation and during the Q&A session. You guys ready? IDP 13, you guys ready?
One of our member is still waiting in the lobby.
Okay. Okay, give me a sec. I'll look into it.
He's still there in the lobby only.
Actually, nobody's there in the lobby. Can you ask him to exit and then rejoin?
Yeah, I'll ask him.
Hello, yeah. Yeah,
You guys ready to start?
yeah, we're good to start.
Okay, start.
All right. So, good evening to one and all present here. So, the problem that was given to us for Parishodhana 2022 was that the CSN capsule, 2 milligrams, is a hard gelatin capsule and it is packed in a PVC blister pack. The product, on 6 months of stability studies, was found to fail in the related substances. After evaluating the pre-formulation study results, it was found that the drug is sensitive to moisture. So our objective was to find the probable root cause and identify the mitigation. The probable root cause for this could be that the PVC, which is polyvinyl chloride, which has been used for the blister packaging of the CSN capsule, it provides a poor barrier against moisture. Hence, the moisture-sensitive drugs, they shall not be packed into PVC. Most PVC sheets for pharmaceutical blisters, they have a thickness of 250 micrometers. Values for the water vapor transmission rate of a 250 micrometer PVC film are around 3 grams per millimeter square per day, which is measured at 38 degrees Celsius, 90% relative humidity. So therefore, hygroscopic drugs and excipients, they tend to lose their stability in PVC blister packaging due to high water vapor rate of PVC. The moisture due to hard gelatin capsules. Now, since we're using hard gelatin capsules, the finished hard gelatin capsules, they normally contain an equilibrium moisture content from 13 to 16% variably. This moisture is, critical to the physical properties of the shells. Since at lower moisture contents, that is lower than 12%, the shells become too brittle and they may even crack when exposed to appropriate stress. So at higher moisture contents, that is greater than 18%, they become too soft and they may lose shape. So it is, important to avoid extreme temperatures and maintain a relative humidity from 40 to 60% when handling and storing such capsules. The bulk of the moisture in capsule shells is physically bound and it can, relatively transfer between shells and its contents depending on their relative hygroscopicity. Conditions, that favor the transfer of moisture of powder contents, they may lead to caking or disintegration or even stability problems. So it may be useful to first, equilibrate the shell and its contents to the same relative humidity within the acceptable range. To ensure the stability and efficacy of the drug due to the moisture content in the hard gelatin capsule, the QC test can be performed, we perform the moisture permeation test. So there are still chances of release of moisture into the content due to the moisture which is present in the hard gelatin capsules, like we told about it initially, and therefore it becomes very essential to avoid the entry of moisture externally into the capsule. Hence, proper packaging for it is very required. Yeah. Okay. So
The mitigation. Polyvinyl chloride is either laminated or coated with various polymers to tackle its problems of poor barrier against
Are
moisture.
these slides?
Guys, you need to change the slide. Do you guys need to change the slide?
yeah, yeah,
Yes,
I
because
thought.
Yeah, you guys need to tell us when you guys need to change the slide.
All right. Okay.
Yeah.
So mitigation. Polyvinyl chloride is either laminated or coated with various polymers to tackle its problem of poor barrier against moisture. So polyvinyl chloride can be coated or laminated by the following polymers. That is polychlorotrifluoroethylene, PCTFE, cyclic olefin polymers, COC, or cyclic olefin copolymers, COC, polyvinylidene chloride, PVDC. So coming to polychlorotrifluoroethylene, PCTFE. PCTFE is modified polyethylene that prevents the entry or exit of moisture from laminated surfaces. Pharmacopoeia standards indicate the lamination of 15 to 150 microns thickness to 250 microns PVC to impart physical protection to packaged formulation. So typical water vapor transmission rate values noted are 0.06 to 0.4 gram per meter square per day, which is less as compared to PVC film, which is around 3 grams per meter square per day. So the PCT FE films have the lowest water vapor permeation compared to all the other plastic films used in blister packaging, and they have thermoforming properties similar to plain PVC, though it is also the most expensive.
Next slide, please. Okay, so the next one is the previous one. So the next one is cyclic olefin copolymers. So lamination of cyclic olefin copolymers is an alternative to PVC as it has improved barrier against moisture. COC also has a high moisture barrier for clear polymer along with a low moisture absorption rate. Its water absorption is less than 0.01%. Water vapor permeability at 85% room relative humidity is 0.02 to 0.04 gram per meter square per day, which is less as compared to PVC film, which is around 3 gram per meter square per day. Next slide, please. So the next one is polyvinylidene chloride, which is PAC. It is a solution to mitigate the barrier against moisture. Its water vapor transmission rate is from 0.65 to 0.25 gram per meter square per day, which is less as compared to PVC. PVDC coated blister films are the most common and prevailing barrier films used for pharmaceutical blister packs. PVDC coatings are also the most economical method for adding water barrier.
Yeah. Next slide. So now we look at we saw the three polymers that we used, and now we look at the most suitable solution. So the most appropriate solution that we use is PVDC, that is polyvinylidene chloride, which is coated on PVC. PVDC coated on PVC, it gives us a moisture barrier which is 5 to 10 times more than PVC alone. So its polarity favors high crystallinity. It provides a dense moisture barrier microstructure. PVDC coating is the most cost-effective method as compared to the other methods solutions, since coating weights can be easily customized depending on the requirements of the barrier. In order to obtain high barrier properties, PVDC is always applied using an emulsion coating process using a PVDC resin dispersed in water. Next slide.
So now we look at the accurate QC test, which is a solution for stability against the content inside hard gelatin capsule. So to ensure the stability and efficacy of the drug due to moisture content in the hard gelatin capsules, accurate QC tests need to be performed. So the Tests related to stability of the drug affected by moisture are determination of moisture content and moisture permeation test. So first we look at the determination of moisture content. So water content of the entire capsule or the capsule content are determined by Karl-Fisher titrimetry to enable the correlation of water content with the degradation profile or drug release characteristics of the capsule. The next one is moisture permeation test. So the USP requires determination of moisture permeation characteristics of single unit and unit dose containers to assure their suitability for packing capsules. The degree and rate of moisture penetration is determined by packaging the dose unit together with a color revealing desiccant pellet, exposing the packaged unit to known relative humidity over a specified time, and observing the desiccant pellet for color change, which indicates the absorption of moisture, and we then compare the pre-test and the post-test weight of the packet. So thank you. This was it.
Okay, thanks. It was a good presentation. I have question only about the last slide. Can you just go back to that slide last? So I did not get why we have this particular section in this problem statement about the accurate QC testings.
Yes, right, sir. So in hard gelatin capsules, we often have problems like they are one of their disadvantages that there is moisture absorption in it. So adequate QC tests would also be required to eliminate those problems, which we overcome by performing these particular tests.
So basically, the finished hard gelatin capsules which are there, they have a certain amount of moisture in it already. So basically, like that moisture goes into the content of the capsule. So if like the content of the capsule are hygroscopic, so they can lose stability. So that is why, and again, if you're using PVC packaging blisters, which is again which like already has low barrier against moisture, so that's why it's very essential to do these accurate QC tests to make sure, it's like an additional thing to make sure about like that the moisture is not.
It definitely acts as a filter, since we mentioned it in the previous slides as well that there is already moisture content present, which is 13 to 16% with hard gelatin capsules. So eliminating that part and along with eliminating the packaging processes, so that is
Okay,
how we
got that one. Yeah. Hello. Yeah, got it. So basically, if I put it in a different way, basically what happens generally, and is that these capsules will be equilibrated with the environmental humidity ultimately. So in a manufacturing setup, we always have fixed humidity in the environment in the production site.
Right.
Okay. So generally it gets equilibrated to that particular level. Okay, so that's one part of it. Second part of it is, whenever you put it into a blister, generally the hard gelatin capsule moisture would always again get equilibrated based on how much is the MVTR of the blister, basically. So if the blister itself is quite having a very high MVTR, would definitely result into absorption of moisture into the capsule shape. But if the material used for blister is good, as you mentioned in your earlier slides, it would ultimately result into a well controlled humidity into the into the capsule shells ultimately, because capsule shells depend on what is the what is the environmental humidity ultimately, and if
Absolutely.
you protect that from the environmental humidity, you're actually protecting the whole capsule. It does not need to be specifically tested and selected one capsule after the other, saying that okay, I want a 11% humidity capsule versus a 16% humidity capsule. So that way it will not work as such. Okay. But yes, it's okay, like from a theoretical perspective, that's good. Yes, that's okay, like you take the best possible option of the capsule available. So that's okay, but there are grades available. You can check for the low moisture grade capsules. Okay. You can change the the gelatin to HPLC capsule, for example, will have a lesser moisture content ultimately. uh, okay, there is one more question I have in terms of selection of the polymer. You mentioned about PVDC. You did not mention about Aclar, but yes, that's one of them. How you would choose out of those three or four? Like you chose ultimately PVDC, but what's the basis for choosing PVDC? It's not only cost, right? Because cost may be less, but ultimately if the product quality is getting impacted, it may not matter what cost it is, because there will be no product ultimately. So how you would select out of those 4 or 5 options you have in terms of different materials?
Right, sir. So definitely we took PVDC as one of the leading materials, because if we even look into the market, in a study, it was found that PVDC at a point was one of the most highly used polymers in the market as well, because it had, when you compare the cost with other things that like PVT, the previous one, the fluoro one. So it definitely has way more cost. That is, approximate cost of it was found to be 71,927 rupees. And when you compare it with PVDC, it costs around 20,900 rupees. So it definitely has a very wide range in cost effectiveness for the packaging materials as well.
Yeah, but,
And
but from a practical perspective, I'm asking this question. It's how you would choose one film over the other. That's my say suppose you have 4 options now here. So for example, okay, cost is one of the factor. Okay, so cost. So is it always you will go for the cheapest one? Then you better stick to PVC.
No, PVDC definitely provides quality as well. It's not that absolutely useless and that is why it's cheap. No,
but
it's
but
not
how
that.
you would choose, like yeah, so how you would choose? That's the specific question. Like
so
how you would
so
choose out
like
of those
we
4?
there are
Yeah,
like
please go ahead.
several aspects we look at when it comes to choosing, like cost effective is definitely one of it, but obviously the efficacy and how effective it is, that is also one of the perspective. So we had mentioned about PCTFE and COC, apart from VCD. So basically, like PCTFE was found to be like extremely expensive. So that was one of the reason to to choose PVDC over PCTFE. And one problem with COC is that it has got very poor resistance with fats and oils. Like it is not related to any moisture content right now, but when we are looking at like from formulation perspective, it is important to check like overall perspective. Like right now we were focusing on just moisture content, but if you look at the overall perspective, we had read this in a research paper that COC had poor resistance against fats and oils. And it was so poor that even the oil which is there on people's hand is like enough to cause the COC resin to stress crack over time. So that is why like multiple layers of COC is required, which again increases the cost. So that is why, like looking at all the aspects, PVDC was found to be the best one. Like it is obviously like one of the most commonly used packaging, like for blister packaging, and it is cost effective, and it was overall found to be the best according to our research that we did.
Okay. Yeah. So I would say the most practical way to choose the material is to actually use it. Okay. Put all these packs into the stability and which one performs better, would be chosen over the other. So that's the most practical way it is done. Otherwise, you cannot just predict saying that okay, this looks cheaper, this looks good in MBTR, so just use it. So that's not the way it would be done. But
No,
yeah,
sir,
okay, good,
like
good, great, nice, nice, thank you.
Sandeep sir, would you
Yeah,
like to
it's
ask any
open for
question?
others. Yeah. No, I'm done with the question. Yeah, yeah, it's open for others. If there are.
Yes, sir. Okay, IDP 13. Thank you so much, guys. Amazing work. You guys can leave.
Thank you so much. Thank you so much.
Thank you. Thank you.
IDP 16, please join.
Yes, Sita, what happened?
No, sir, in case if you wanted to ask any questions. IDP 16, you guys need to keep your camera on throughout the presentation and Q&A session. Are you guys like ready? Yeah. Should we start? Yeah, you guys can go ahead.
So yeah, can I start? A very good evening to everyone. Our problem statement is as follows. A wet granulation based product manufactured at a European site was being transferred to a manufacturing site in Vishakhapatnam in August 2020. The granulation process was based on a top spray fluidized bed granulation, although the machine utilized at both sites was identical in terms of size, capacity, geometry, number and type of spraying guns, etc. However, there were few controls which were missing. Example, the online data backup, dew point control, etc. In the first batch, it was observed that although the parameters were strictly controlled during granulation, the granules formed were coarser as compared to the parent site, and this impacted the dissolution of the product. Our objective was to identify the root cause and suggest a mitigation plan. Next slide, please. The problem of coarse granules may occur due to lack of proper controls like the dew point control, online data backup, etc. The relative humidity in Vishakhapatnam in the month of August is an average of 81% as compared to that of the European continent, which is 66%. Both these factors contribute to the change in the quality of the granules and hence the dissolution. My teammate will now continue, and could we have the next slide, please?
Humidity as an external factor can have serious effects in the process of granulation. When the relative humidity exceeds a critical value, the absorbed liquid layers on the particle surface can enhance the interparticle forces by formation of liquid bridges between the particles, making them coarse. The relative humidity increases the cohesiveness and thus influencing the mixing behavior and the granule growth. An increase in relative humidity leads to a decrease in the duration of stable operation. There is degradation of the fluidizing quality due to higher local humidity, which may further amplify interparticle adhesion forces. Next slide, please. Another important aspect is the inlet air. as it has an effect on parameters such as temperature and humidity, which greatly affect the quality of granulation, coating, and drying. When the outside humidity is high, dehumidification of the inlet air has to be maintained at a specific dew point. However, due to lack of dew point control in the present problem, the humidity and temperature of the air are not maintained. Relative humidity of the inlet air also affects drying. The drying capacity of the air depends on the relative humidity of the incoming air. At 100% relative humidity, the air is holding the maximum amount of water possible at a given temperature and is not able to carry the evaporated water from drying. This may lead to agglomeration due to insufficient drying. Ensuring that the prescribed storage conditions are adhered to is also very important in this situation. Can we have the next slide, please? Before we move on to the solutions, let us have a brief look at the basic principle of operation of a fluidized bed granulator to have a better understanding of the process modifications. Initially, the product is loaded, followed by passage of treated air, and then the fluidization begins. Spraying commences from a top spray with simultaneous addition of moisture and evaporation of water. After spraying is completed, drying continues gradually, and finally the granules are discharged. Could we have the next slide, please? Our mitigation plan includes the following solutions. Firstly, the installation of filters. Distinct heating or cooling sections can be installed in an air handler depending on the geographical location of the plant. Since the unit in question is located in a tropical and humid climate, the humidity removal section is employed first. The second is to choose an inlet air temperature that is high enough to negate the weather effects like the outside air humidity or the inside room conditions. The air temperature should not be detrimental to the product that is being granulated. On to the next slide, please. Another method that could be employed is the use of a bypass loop. A bypass loop can be used for reconditioning the air. This loop allows required process temperature and humidity to be attained within the system ducts before the product is subjected to fluidization. Yet another method is to dehumidify the air to a preset dew point to maintain incoming air at a constant drying capacity and hence provide reproducible process times. Next slide, please. The various methods to dehumidify air and lower the dew point are as follows. The first method is to subcool and reheat. Subcooling followed by reheating is the most common method of dehumidification. This method involves cooling the air to a desired dew point temperature, then reheating it to the desired temperature. Passing air over a cooling coil whose surface temperature is below the dew point of the moving airstream causes condensation, which drips off the coil and runs down a drain. Air leaving the cooling coil has a lower moisture content than before, but it is cold and hence has to be reheated to raise its temperature and lower its relative humidity as per requirement. Before this process is done before it is delivered to the system. A second method of dehumidification uses a desiccant wheel. Desiccant dehumidification makes use of the property of some chemicals such as silica gel and lithium chloride to absorb moisture from the air at moderate temperatures and releases that moisture when heated at high temperatures. Desiccant dehumidification systems typically involve passing air over a moving honeycombed wheel containing the absorbing chemical. As the wheel rotates, it passes through a separate stream of heated regenerated air that is vented outdoors. This regeneration stream causes the chemicals to release the moisture so that by the time the wheel completes a revolution, the chemicals are ready to absorb new moisture again. Could we have the next slide, please? Yet another method is that to employ near infrared spectrum to determine the quality of granules. The near infrared spectrum is just above the visible region of the electromagnetic spectrum. The NIR region covers intervals between 0.8 to 2.5 micrometers. This band causes the OH bond stretching and bending vibrations and is most useful in analysis. The water content has an effect on the NIR absorption at all wavelengths, even when water is being absorbed. It can be used to study granule moisture measurement combined with temperature and humidity measurements, and the varying behaviors of formulations during processing can be identified in real time. This process can be used to identify an end point of the granulation and prevent agglomeration and further processing. Could we have the next slide, please? And over to my teammate for further explanation.
Other formulation-based changes include increasing the quantity of disintegrant or decreasing the quantity of binder. A disintegrant is an additive that promotes disintegration, which is the breakage of a tablet into small fragments when in contact with a liquid medium. By increasing the amount of disintegrant added to the formulation, the surface area available for dissolution is increased and drug dissolution is accelerated. On the other hand, the primary role of binders is to provide cohesiveness essential for the bonding of solid particles by enhancing intragranular and intergranular forces under compaction to form a tablet, while also promoting size enlargement to produce granules. Hence, reducing the amount of binders in a formulation may increase disintegration time and does increase dissolution rate. Another solution is size separation of coarse granules. Agglomerated granules can be separated out by various size separating apparatus. The particles of optimum size can be used and coarse granules can be further processed. The further processing can be done by size reduction of coarse granules. Reduction in size causes both an increase in contact area and decrease in disintegration as well as dissolution time. Increasing drying time can also compensate for the excessive humidity in the environment. Maintaining humidity and temperature of the warehouse is also important as it directly affects the moisture content and quality of raw materials. Hence, good warehousing practices must be employed. Keeping identical parameters in places with different climatic conditions and expecting identical results is a challenge, and hence, making the necessary rectifications in the process, formulation, or equipment is important. The mitigation plan thus suggested shows promise in overcoming the problem in the best possible way. Thank you.
Okay, thanks a lot for this presentation. It was nice, and I agree that the root cause which you identified is the right root cause, I would say, in this case. No, my point is only with respect to the last two slides wherein other multiple options were mentioned, specifically with respect to the formulation. But if you see the problem statement, it talks about transferring it from one site to other. It talks about reformulating the product as such. Okay, so we need to focus only on how to solve the issues at the plant rather than changing the formulation, because ultimately, yes, formulation change is always an option, but it's not in this case, at least, because we are actually transferring the product from one site to other. This is not a new product; it's already there in one plant, and we are just transferring it to the other. Okay, so that's one comment. The otherwise, I agree to most of the points which you presented. Only one question with respect to you mentioned two options. One is, I think, the dew point control, which you need to go put into place. But if if there are some concerns, say the plant where the product is getting now manufactured is a third party. So, for example, it's not the own plant of that company, and they are giving that contract to a CMO, contract manufacturing organization. And there are concerns with respect to the investing so much amount into modifying the equipment. What can be the option to solve this issue?
I would like to answer this question. So as we already
if
mentioned,
we don't put the dew point control.
Yes, sir. So as we already mentioned that we can either change the dew point control or we can increase the drying time. As we all know that drying is directly proportional to the relative humidity present in the atmosphere. If the relative humidity is higher in the site Visakhapatnam, then if we increase the drying time by X amount, we can compensate the excess of atmospheric humidity present. So if we increase the drying time, maybe the rate at which we can get sufficient drying after increasing the drying times.
Okay. Yeah, but yes, that's one. But that's not a very specific answer. I would say, the drying time is basically talks about removing water at the end of the process. Okay, and whenever you are granulating, it's the whole process is important. And being a fluid bed, again, it's not a high shear granulation. It's basically a fluid bed, which actually works on the equilibrium between the spray rate and the drying rate. Okay, so you are putting spray solution onto the excipient mix, and you are drying it at the same time with the fluidized air at a certain temperature. Okay, so this is a balance between both of these. So there is a balance between the spraying amount and the drying amount. Okay, so you need to put that balance in mind. So basically, the simplest way would be slightly increase the inlet temperature, because the RH is basically a function of inlet air temperature as well. Yes, the RH is basically the function of two factors. One is the water content, which is out of our hand because it's Vishakhapatnam, and in August you will not be able to reduce that ultimately. But you can always increase the temperature, which would effectively reduce the RH overall. So that's one, and increase would not be like in terms of 10 degree or 15 degrees. It would be just 2 degree to 3 degree. You can check that there is a chart for RH and temperature conversions. You can find with an increase of about 1 to 2 degrees how much difference in the RH happens to the air. So that would help you to decide what's the actual temperature needed in that particular situation without investing into a dew point actually. Okay, so good, otherwise nice. I agree to most of the points which you presented in this presentation. One more suggestion is that try to avoid more details. Many a times those are not required. It was a specific problem, we stick to that. You presented somewhat more details of many other options which may not be directly related to this particular solution, but yes, you can reduce the number of slides as such. Okay, that's just a general comment, not related to this competition as such. Okay, thank you, thanks a lot. It was nice to have all those options in front of us for the same
Thank you, sir.
problem. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Could we leave the meet?
Yeah, you guys did a great job. You guys can leave the meet.
Thank you.
Thank you, sir.
Thank you.
Thank you.
Next is IDP 17. IDP 17, you guys will have to keep your cameras on throughout the presentation and the Q&A session. Are you all ready?
Yes, we are ready.
All your members are here?
Just a second, I will check.
Yeah, sure. You guys ready to start? Yeah, take your time.
Yes, we are ready to start.
Okay, guys. You guys can start then. Let's go.
Okay. Good evening to one and all. We
2017 are here to present a solution to the given problem statement on the topic of nasal decongestant on the event of Parishodhana 2022. So first, let us have a look at the problem statement. A nasal formulation is development. It was observed that the formulation residence time in the nasal cavity is not sufficient to produce its pharmacological action. So for this purpose, our objective was, what are the options to improve the residence time in the nasal cavity? Next slide, please. Let us have a look at the basics of nasal decongestant. Talking about its definition, it is a type of pharmaceutical to relieve nasal congestion in the upper respiratory tract. It is used into the patients like those suffering from allergic rhinitis. Allergic rhinitis is nothing but inflammation of the inner part of the nose. The class congestion can of action into three types as given below. That is short-acting decongestant administered topically, long-acting decongestant administered orally, and long-acting decongestant administered topically. Next slide, please. Focusing on the objective, so we had to overcome these limitations. So in order to overcome the limitations of the given nasal decongestant spray formulation, we focused on a technique. This technique helped us to improve the residence time in the nasal cavity. So our technique which we opted was nano suspension based in situ gelling nasal spray. Next slide.
Moving towards the methodology, the opted method of nanosuspension improve the bioavailability and therapeutic efficiency of formulation. When we prepare this nanosuspension, ultrasonication further incorporated in the gel display. When this in contact with gel fluid convert to gel was produced via the ion activated gel and gum as per here. These are some ingredients Queen 80, methanol, ethanol.
Next slide, please.
Here preparation is explained in stepwise manner. First, preparation of drug stabilizer solution in organic solvent. Drug stabilizers such as Queen 80, Span 40 or combination can be used and organic solvent like ethanol and methanol. In second step, this drug stabilizer solution is added to pre-cooled aqueous solution like deionized water with simultaneous application of ultrasonication using probe sonicator, followed by evaporation of organic solvent. This gives drug nanosuspension. Further stable drug nanosuspension is mixed with in situ gelling polymer dispersion. Here, gel and gum can be used as in situ. Resulting solution filled in. Next slide. Followed by gelling. Final in situ other ingredient nasal cavity. Comes to physiological changes like temperature or nasal concentration. The conversion side this gives benefit like high retention, bioavailability, stability, avoid barrier, rapid.
Next slide, please.
Talking about the advantages which can be offered by our formulation include First, in situ gel forming system have certain advantage like increasing retention time in nasal cavity and also show permeation enhancing capability. Second, benefits obtained from the both solution and gel by an impressive number of in situ gel forming system that are induced by temperature, pH and concentration, ion concentration been prepared from the use in nasal drug delivery system in past few years. Third, nano suspension also provide high bioavailability and dose regulation. Fourth, it also reduce the dose frequency and hence increase patient compliance. Fifth, in addition, it also prevent mucociliary clearance and enzymatic degradation. Next slide.
Following evaluation can be done for the formulation. First one is the pH study. pH for the formulation can be determined using calibrated digital pH meter. Second one is the appearance. Appearance can be identified visually for the clarity in the solution and gel under black and white background. Third one is the gel strength. Gel strength is a measure of the ability of a colloidal dispersion to develop and retain a gel form. It can be performed using the gel strength apparatus. The picture on the right hand side is the apparatus for the gel strength. Fourth one is the mucoadhesion strength, mucoadhesive strength. Mucoadhesion is the adhesion between two materials, at least one which is in the mucosal surface. A modified balance technique can be used for determining the mucoadhesive strength. The picture on the left hand side of the balance is the mucoadhesion strength apparatus. Next slide. Next slide. Lastly, these are some of the references which we referred for this presentation. Next slide. Thank you all for your patient listening.
Thank you for this presentation. So if I understand correctly, the approach you chose to increase the residence time is in situ gelling, right?
Yes.
Hello.
Yes.
Okay. Okay. And what is the reason behind using nano suspension as a technology in this case? If we look at the problem statement, what's the rationale using nano suspension?
Nano suspension had an added advantage that it not only was helpful in increasing the residence time, but also it was useful to increase the bioavailability of our formulation if used. Therefore, we opted for nano suspension in situ gel system.
But is the bioavailability was a problem in this case?
No, we had to increase the residence time. That was the main aim. But by increase of the bioavailability was the added advantage by using nano suspension.
Which polymer examples you can use for this in situ gelling system?
Here we use Galan gum as polymer for in situ gel formation.
Any other options you have?
Chitosan is also used, and carbapol, HPMC are also used, can be used for in situ gel polymerize.
Yeah, they're gelling, mucoadhesive gelling agents.
So for in situ, in this case, any other polymer you have as example? So the one which you gave are more of like a general gelling agent. So basically, once you put it into the water, HPMC would always form a gel. It's not in situ or something like that. It's a gel. So any other example? Okay, what's the mechanism of gelling in this case? Say Jalangam. What's the mechanism of gelling, in situ gelling in this case? Once it's sprayed already onto the
In given example, galangum is gives response with the ion concentration. In note, the ion in response to ion concentration, it forms complexation with ions in the nose mucosa and forms gel.
Okay. Okay. So are there any specific ions required, or it's there is some specific osmolarity which is required? How you would evaluate, in situ it will form sufficient viscosity gel or not?
Using the evaluation parameters which we selected, like gel strength, we can evaluate.
No. Yeah, that I got. My question is, how the in situ part of it would be evaluated. I'm more, see, gels are okay, like, you can measure gel strength and all those things. But, when you say in situ means it will form inside the nasal cavity. So would it effectively form a gel inside the cavity or not? How you will evaluate that? That was the question.
In rhinitis, nose muscles get inflamed. So in there, the concentration of ion will increase, like sodium, calcium, or magnesium. That increased ion concentration will trigger the polymer to form complexation and gel form.
What would be the normal concentration of ionic concentration in the nasal cavity? And what would be the increased one in rhinitis? Okay, during your brainstorming, did you think of any other option other than, the in situ gelling for increasing the residence time? Okay. So one of the option is, using the thixotropy. Okay, so thixotropy is, the property of gels. They reduce in viscosity when you apply stress. Okay, so those type of polymer would also help in this case, because, when you spray, you actually put a mechanical stress which will reduce the viscosity of the spray, and then again forms a gel inside the nasal cavity when utilized. So that's one of the option, and you can, dig further, from that concept as well.
Yes.
Okay, so I think I'm done with my questions.
Okay.
Okay, I think then we will take another presentation.
Okay, sir. Thank you guys for your presentation. It was amazing. You guys can
Thank
leave the
you.
meet.
Okay.
Thank you.
Thank you.
So Sripas sir, it's very hectic for you after 3 hours.
Now we have the last presentation, that is IPD 06, please.
This is last one, na?
Yes, sir, this is the last one.
We would have been offered some tea or other things if this event is on offline mode. This is IDP 06.
Yes, sir. Hi guys, IDP 06. You guys need to all keep your cameras on for the presentation and for the Q&A session. Let me know when you guys are ready. Okay.
Okay, we can start.
One more team member needs to join, I think.
Okay.
You guys ready?
One team member is unable to join. She has connectivity issues, I think.
Okay.
So you can start. Whether she is the first to talk.
No, she is not.
So if you wish, you can go ahead.
Oh, okay, sir.
What is her problem?
She has
Just ask.
connectivity issues, sir.
रिजॉइन बोलो ना उसको रिजॉइन करने को. Let us see. Okay, connectivity issues there. You can continue.
Yes, sir.
Or maybe you can. Any one of you can take her part. That thing is also possible.
Yes, sir. Yes. We are just informing her regarding this.
Okay.
Yes,
Okay.
Yes, sir. We will start.
Yes. Yes.
So, okay. Good evening to one and all present here. This is code IDP06. The problem statement given to us is CSN capsules 2 mg is a hard gelatin capsule packed in PVC blister pack. The product on a 6-month stability study was found to fail in the related substances. After evaluating the pre-formulation study, it was found that the drug is moisture sensitive. To know in detail about the problem, we refer to the formulation manufactured by Shreya Life Sciences Private Limited. Please share the next slide. Hello. To understand this, we need to know what CSN capsules are. CSN capsules are the unique combination of multivitamin nutritional supplement and is recommended as adjuvant therapy. Its composition includes coenzyme Q10 10 mg, docosahexaenoic acid 60 mg, icosapentaenoic acid 90 mg, folic acid mg, mecobalamin 500 microgram, vitamin B6 3 mg, vitamin C 150 mg, and vitamin E 200 mg. Please change the slide. So, what are the problems and the probable root cause of this issue? The product on 6-month stability study shows failure in related substances. This can quite be possible due to the moisture sensitivity of the drug. Moisture has a real impact on the formulation stability and can also chemically degrade the drug, which is why the product fails in the relative stability studies. The following problems need to be resolved in the and should be improvised in the formulation. First is hard gelatin capsule shell, and second is PVC blister packaging. Hard gelatin capsule shell. Please change the slide. It contains 13% to 16% of moisture content, which is necessary for maintaining its physical properties and its rigidity. At lower moisture content, that is less than 12%, the shells become brittle and may crack. This relatively high presence of water is unsuitable for moisture sensitive drugs, and hence there is a need to change this the capsule shell because of the changing physicochemical properties and thus leading the drug towards the degradation. Now, please change the slide. PVC blister packaging. These are the most widely used material for blister packagings. Its main disadvantage or limitation is poor barrier against moisture ingress. It is not suitable for moisture sensitive drugs. This also has relatively low shelf life, and it also negatively impacts the environment as it releases CFCs. To tackle these problems, we propose our formulation with relevant solution. Please change the slide. We propose that hard gelatin shell should be replaced with HPMC shell, which are more efficient and has low moisture sensitivity and can contribute in enhancing the stability of moisture sensitive drugs. The next is the packaging material, which can be improvised with addition of desiccants, which stabilizes the internal environment of the packaging, thus establishing a longer shelf life of the product. Over to my teammate.
Can you please change the slide? Okay. So coming towards the selection of the HPMC as a capsule shell, it is primarily selected for its physical stability in low moisture content and minimal reaction with the water. This can be explained as follows. Brittleness is a critical aspect when capsules comes into play. Water in gelatin capsules functions as a plasticizers, and when this level falls below the specification limits, the capsules becomes brittle and cracks on handling. However, in the HPMC capsules, water does not act as a plasticizer, and hence the moisture content can be decreased while maintaining the mechanical strength. So, compared with the difficulties found in the hard gelatin capsule shell, not only is the hardness of the capsule maintained and has relatively low moisture content, but it also gives other benefits like low brittleness, decreased use of the excipients. Also, they are obtained from the plant sources, so less preservatives and chemically inert formulations. Can you please change the slide? So further modification in the material processing is the HPMC extra dry capsule. So this can be achieved by optimizing an additional drying step which do not affect the physicochemical properties of the HPMC. With this technique, a formulation with very less moisture content is derived, that is about 2 to 3.5%. Thus, it ensures the solution to the moisture sensitivity of the drug. Also, they have found in the stability studies that the moisture content of the HPMC extra dry capsule was about 2 to about 3.5%. Also, the capsules are less brittle, then they are stable at the storage conditions as well as handling conditions, and also the shelf life of the final drug product was found to be about 2 to 3 years. Over to my teammate.
Please change the slide. Yes. So coming to the packaging solution, the most of the drugs are highly sensitive, meaning they may exhibit undesirable chemical, physical, or pharmacological changes when exposed to moisture, particularly in humid environment, leading to ineffective or even unsafe products. The traditional cold form packaging solutions lack a critical functionality that is optimum moisture protection. As a result, the manufacturers need to incorporate desiccant in some form into their packaging. So to overcome this problem, we have the integrated desiccant blister packaging, which is a cold form blister packaging solution with an integrated desiccant in the sealant layer, protecting each blister individually and thus enhancing consumer safety by removing the risk of desiccant consumption. The major advantage are the accelerated speed to market. A pharmaceutical product's success is largely determined by how quickly it gets to the market. So to achieve the full-scale production in less time has its own competitive advantage and financial benefits. Using this packaging solution, it can accelerate stability tests and reducing costs and increase speed to market by 5 to 10%. Also, it has improved barrier function. The data shows this form of packaging can delay moisture ingress into the blister cavity with relative humidity less than 10% for over 5 years, even in hot and humid climates, compared to standard cold form blister solution. The highly effective desiccant integrated into the sealing layer, which reacts chemically and irreversibly with moisture, thus significantly increasing protection against cross diffusion and totally ensuring product stability. Over to the next slide. Please change the slide. Please change the slide.
It's changed. It's changed. Please change back to previous one.
Yes. It has the integrated desiccant blister packaging has excellent blister machinability and long-term resistance to delamination. Also, the lower packaging cost, increased shelf life due to more effective pharmaceutical packaging, thus decreases the number of drugs expiring. Avoiding patients' consumption of out-of-date drugs and reducing waste of drugs. The manufacturers can also avoid the complexity of adding components such as pouches, overwrapping, or separate desiccants or cavities into the product. Also, the reduced carbon footprint, such as that is the better sustainability and environment friendly as it is PVC free packaging. PVC is the most environmentally damaging of all plastic. Its production, use, and disposal results in the release of toxic chlorine-based chemicals and dioxins. So next slide. So the advantages of our proposed solution are our proposed solution is suitable for moisture sensitive drug. It has better stability, improved shelf life, and it is environment friendly. Next slide. Thus, our solution is more effective with significant cost savings and long-term competitive advantage. Thank you. We are done with our presentation.
You are on mute, sir. Sripad sir, you are on mute.
So we have presented two solutions here. One is with respect to the capsule shell, and the other is with respect to the packaging material.
Yes, sir. Yes.
So in case of packaging material, if I understand correctly, it's already ready to use packaging material with desiccant inbuilt into it, right?
Yes, sir. Yes.
And it's alu pack basically.
Yes.
So why you chosen this particular material over the others?
First of all, since the problem statement stated that the earlier product has PVC packaging, so first we found out that the disadvantages of PVC as a packaging. So since PVC does not provide the optimum moisture protection, so we were in search of some other alternative to this kind of packaging. So there were other cold form packaging, but also it has various limitations. So in the end, we found this as the best possible as a packaging solution with this many added benefits of accelerated stability tests and lower packaging cost, etc. So that
I
is
would
what are the
also
other
like to add on
options?
this.
Yeah,
Like
please. Sorry.
yes, so in this blister packaging, in this desiccant packaging, we also found alternative as bottle canister packaging, which also included desiccants and silica. But in case of bottle packaging, though the internal environment of the drugs, the internal environment of the packaging is maintained at a lower relative humidity. But since we are going to open it again and again for consumption of the medicine, it can again affect the moisture content. So instead of using bottle, we used the blister packaging.
Okay, in blister, how many other options you evaluated other than yes, I know you showed a disadvantage with the PVC, but what other options you had when you chose ultimately the one with this?
We had the aluminum foil blister packaging. That was one of the options.
And why this that was not chosen?
Sir, in that the desiccant was not there. So we thought that since our product is ultimately has to be moisture protective, and also that does not provide that other added advantages. So that is why we chose this. Also, the traditional cold forming packaging also uses PVC as a material. So that's why we didn't choose that.
There are also the over the period of time, the maintenance of relative humidity was observed lowest in the type of blister packaging where desiccants were used. Even in allulo blister packaging, it was slowly rising the moisture content relative humidity.
Okay. So just to give perspective in this, you have to think about ways on how you will choose a particular material. Okay. So okay, this may be one of the option, but this may be one of the costliest option as well. Okay. From a product development point of view, you need to balance between all of these. It's not only the best material. Most of the time, it's best, which is I would say fit for purpose material. It's not always the best material which needs to be chosen. Okay, so it has to be fit for purpose. So what's the purpose is that you want to stabilize the product. Okay, a simple alu would be probably a sufficiently providing sufficient barrier for the moisture, uh, because traditional alu is basically a metal film ultimately, so it will not as such allow unless and until your sealing properties are not good. Okay, so you need to consider all of the options. In blisters there are multiple films available which can be evaluated basis MVTR, moisture vapor transmission rate, and you can even do some studies on your product stability studies and choose one out of them. It's not always just going best case scenario. It may not be always the best case material is required. It may be lower than that, or two level lower than that as well would do. Okay, that's one. Secondly, I saw you mentioned a lot about the capsule shell moisture content, and I agree to some extent with respect to the HPMC capsule shells, that you may go with those to reduce the inherent moisture into the product. Uh, but generally, the capsule shell moisture is very limited to the, and it's very dependent on the environmental moisture ultimately. Okay, so when you actually manufacture a product, it equilibrates with the environmental moisture, and in the production unit, generally the moisture is already controlled. But once it is put into the blister, it's basically the blister MVTR, which decides how much capsule shell would take up moisture. From a theoretical perspective, yes, HPMC would give some benefit because the inherent moisture is less. So I agree to that from a theoretical aspect, but from a practical aspect, many a times it's not very effective in curbing the moisture sensitive drugs degradation. Okay, it's more of a film which would decide how much moisture would go inside, and then would protect the product or not. There is no product, when we gave this particular problem statement, CSN, you must have found it somewhere in Google. It's more of a example name. It's not, there is no such product called CSN tablet or capsule with so and so. There may be some similar brand